about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Live that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Live experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Live, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Live that you can't see, and I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Live experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Live, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Upside down. Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on. I stand tall. I stand tall. Head held high.
I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall. I stand tall, my life grows. I stand tall. I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall. I stand tall, my life grows. I stand tall. I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall. I stand tall, my life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLife2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What?
Upside down.
Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see, and I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall. I stand tall, my life grows. I stand tall. I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall. I stand tall, my life grows. I stand tall. I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall. I stand tall, my life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLife2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. I'm watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on. I stand tall. I stand tall. Head held high.
I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall, I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall, I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Upside down. Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on. I stand tall. I stand tall. Head held high.
I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall, I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall, I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLife2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What?
Upside down.
Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall. I stand tall, my life grows. I stand tall. I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall. I stand tall, my life grows. I stand tall. I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall. I stand tall, my life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLife2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What?
Go ahead.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Live experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Live, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What?
Upside down.
Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see, and I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on. I stand tall. I stand tall. Head held high.
I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall, I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall, I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall.
Welcome to MS Live 2014. I'm with Shaa Pizaro, who is going to be doing a talk, Escaping Labels. Hello, Shaa.
Hello.
Could you tell us a little bit about the talk and what you're going to be talking to people about?
Well, I'm going to be talking about a postcard that I entered into a project called Postcards from the Edges. And the brief was, what would you want to say to the world about being disabled? Put it on a postcard. And I sort of laughed, and there was a picture of me smoking in a back alley, and I wrote, I drink, I smoke, I swear, on it, sorry, and sent it off. It got a lot of press attention, and it's been, travelling around the country in an exhibition and ended up in Parliament. Fortunately, I know a lot of politicians quite well, and just said, oh, it's what you'd expect from me, and they laughed. So, so I'm talking about that, and I'm talking about why I did that postcard, and actually what's behind it in terms of dealing with a diagnosis with MS, coming to terms with being disabled, and the way that MS affects my life, and the way that I've kind of overcome that to make sure that I still get to be me.
Now, the talk you're doing is Escaping Labels. Could you talk to me a little bit about what labels you're thinking of escaping?
Well, I talk a bit about how disabled people are represented in the media, because I think that very much there's sort of three categories that the media present disabled people in, and one is Paralympian supergods. The second is liars and scroungers and benefit cheats, and the third is sort of tragic cases that need to be pitted. And I don't think I'm any of them. And I don't think most disabled people are any of those. And so, yeah, I want to talk a bit about that.
Also in the talk, it's talking about low self-confidence. Could you give some tips about low self-confidence and how to overcome that?
Yeah, I was in a marriage to someone who couldn't cope at all with the diagnosis of MS, and he very vocally would tell me that I was disgusting because I was disabled, and that he couldn't find me sexually attractive, knowing that I had to catheterize. He was really very abusive, and in fact my divorce got put through as a priority case on the grounds of abuse. I knew that I couldn't handle that myself, and I couldn't see how I could ever sort of regain my self-esteem and my self-confidence without getting professional help. And so I went off to a counsellor, and I found the counsellor that was right for me, and I spent 6 months with him. And at the start of it, I said, I want to believe that I'm still me, and that I am sexy, and that I can, still be found attractive. And through seeking that professional help, that was the thing that really changed things for me. So I think that would be my real advice to people, is seek out and accept the help that you're offered.
Thank you very much. And I think everyone could agree that Shana's looking very sexy today.
Good morning, ladies and gentlemen. My name is Nick Riker, Director of Policy and Research at the MS Society. Before I start, we have had a fall in the room, and I just want to check that it's appropriate for us to continue rather than wait. Yeah, okay, good. Thank you. This morning is going to be a fascinating discussion. Hopefully you would know that over a number of years now the MS Society has been investing pretty heavily in myelin repair, crucial area in terms of the breakthroughs we're all looking for in MS research. Some of you will have heard the conversation yesterday where we talked about one day arriving at a state where we'll have combination therapy. Some of the therapies that people today are able to benefit from, coupled with therapies around myelin repair and neuroprotection, that together could form really effective combination therapy for MS. So MS could then be managed a bit like HIV is today, much more effective, where people don't have to live in so much fear of what might happen to them, that they, with hope, wouldn't then be experiencing the range of symptoms that people typically experience today. So that's what we're really gunning for. Crucial to that is the work that we're doing around myelin repair. We've invested very much in work being done at Cambridge primarily, but also Edinburgh. They've been working very closely together to push this forward. And I'm delighted to say that today we have, I would say, one of the world's leaders in terms of myelin repair, Robin Franklin. Robin has a really interesting background, which I won't necessarily go into now. He may well tell us, or you could ask him. Fascinating as to how people form their medical careers and the paths they go through to get to where they are. Robin is Professor of Stem Cell Medicine at Cambridge. He works in an institute funded by the Wellcome Trust and the Medical Research Council. He's also head of translational science. And very importantly for us, Robin is director of the Cambridge Centre for Myelin Repair, which is funded by the MS Society. So Robin, if you'd like to talk to us, we'll then have plenty of time for questions and answers. Thank you.
Nick, thank you very much for that kind introduction, and thank you very much for coming along this morning to hear what I have to tell you about the work that we've been doing in my centre in Cambridge and also in collaboration with the Edinburgh Centre, which is co-directed by Professor Charles French-Constant, Professor Siddharthan Chandran. So, this is what is in store for us over the next 45 minutes or so. So I'm going to try and provide some answers to the following 4 questions. Firstly, describe to you what myelin is, and more importantly, what it does. Secondly, describe exactly what we mean by the term myelin repair, or more accurately, the term remyelination. These are synonyms, but and I'll explain what they mean in just a moment. Then thirdly, why myelin repair is a very important objective in this sort of comprehensive, multi-component strategy towards for MS therapy. And then finally, and perhaps of most interest to most of you, where we're up to in our joint centres in identifying ways that we can therapeutically make this regenerative process more efficient in the clinical context. Okay, so let's kick off with the first of these questions, myelin, what it is and what it does. So here is a diagram which I've used to introduce my talk for many years, and the reason I've used it often is because it's very simple and a very, I think, clear way of describing the key components of the story that is going to unfold. So what you see here are the crucial cellular constituents of the central nervous system. The central nervous system is the term we use to describe both the brain and the spinal cord. So what you see over on the left of this diagram are 3 grey circles, which are nerve cells or neurons. Nerve cells and neurons have axons or nerve fibers, which head off some considerable distances often to connect with other nerve cells or neurons, and thereby construct this extraordinarily complex circuitry, which is characteristic of the human brain and spinal cord. In fact, it's probably the case that the wiring of the human brain is the most complex structure in the known universe. Not that we know everything in the known universe, but, well, no, we do know everything in the known universe, but not in the universe. Anyway. So the other cell type you see there is the green octopus-like cell at the top of the image. And this is a cell that we call the oligodendrocyte. There's no simpler term for it. It's the oligodendrocyte. And the oligodendrocyte is responsible for making the myelin sheath, which surrounds and wraps the nerve fiber or axon. And that's perhaps illustrated more clearly in the following diagram. So here in the middle, you see the oligodendrocyte, a cutaway diagram of that important and crucial cell. And you can see that there is a central component called the cell body with a nucleus within the middle, which is a dark brown structure. And coming off from the oligodendrocyte cell body are short processes which end in large sheets of membrane which wrap Swiss roll style around the axon. And that constitutes the myelin sheath around the nerve fiber. And you can see that a single oligodendrocyte can make many individual myelin sheaths. In fact, a single oligodendrocyte can make up to 80 separate myelin sheaths surrounding 80 nearby nerve fibers. Here is what the myelin sheath actually looks like when viewed by electron microscopy. So this is looking at the myelin sheath of a rodent. This is many tens of thousands of times magnification. And here you're looking at a nerve fiber cut in what we call transverse sections. So this is like slicing French bread. So in the middle, the pale area is the nerve fiber axon, and the dark staining structure around the outside of the axon is the myelin sheath. And you can see these concentric rings of dark staining, which are formed by the wrapping of the membrane of the oligodendrocyte around the nerve fiber to form the myelin sheath. So what does the myelin sheath do? Well, there are two key functions of the myelin sheath, and it's important to understand these to understand why symptoms arise when the myelin sheath is lost and damaged. The first function of the myelin sheath is one that we've known about for many years, and that is the function of conferring on the nerve fiber the ability to conduct electrical impulses much more rapidly than would otherwise be the case. So the way that nerve cells communicate with each other is by way of electrical signals which form a continuous conducted impulse down the nerve fiber. And as a result of the myelin sheath, for reasons that we don't need to go into any detail, that process is made very much quicker than would otherwise be the case. So because of the myelin sheath, the action potentials, these electrical signals, can whiz down the nerve fiber with some speed. And if you do the maths, you can figure out that in terms of something that's perhaps easy to understand, in the absence of a myelin sheath, the electrical signals would travel at the speed of a tortoise, but in the presence of the myelin sheath, the electrical signals travel at the speed of a fast-running cheetah. And those of you who know something about the animal kingdom, which I assume is all of you, will know that cheetahs are very much quicker animals than tortoises. So you can see that in the absence of the myelin sheath, the electrical signals are proceeding very slowly. In the presence of the myelin sheath, the electrical signals proceed very rapidly. Now, the second important function, and perhaps in some ways the more important function when one thinks of the long-term implications of the disease, the second important function of the myelin sheath is that it protects and in some way nourishes the underlying nerve fiber. And here the best analogy is probably the lagging around the hot water pipes in your house. So in the presence of the lagging, the hot water pipe is protected, so you can have a cold spell and you know that your hot water pipes are going to remain intact. But if you don't have lagging around the hot water pipes and you have a cold spell, then the hot water pipes can snap and break. And just as the breaking of nerve fibers in the central nervous system, those hot water pipes won't spontaneously regenerate. In fact, in the central nervous system, if a nerve fiber breaks, it's all but impossible to get that nerve fiber to regrow. So preventing the loss and damage of a nerve fiber is a crucially important thing to try and achieve in any clinical situation where there is potential vulnerability of the nerve fiber in the central nervous system. So those are the two key functions of myelin in the central nervous system. One is to allow electrical signals to proceed much more rapidly, and secondly, the myelin sheath protects the underlying axon, preventing it from being damaged or, frankly, snapping. So that's the key constituents of the central nervous system, the nerve fibers and their axons, myelinated by an oligodendrocyte, which makes myelin sheaths around the nerve fibers. Now, MS is a disease characterized by a disturbance of the immune system, such that the immune system gets itself confused and starts to imagine that components of the central nervous system shouldn't be there, and the specific component that is targeted in multiple sclerosis is the oligodendrocyte and the myelin sheath that it makes. So you have an immune system which goes awry for reasons that are still far from clear. It kills the oligodendrocyte. As a result of the death of the oligodendrocyte, the myelin sheath comes off the nerve fiber, and the nerve fiber is no longer myelinated. And of course, that'll have immediate implications for the ability of that nerve fiber to conduct electrical signals, and will eventually have implications in terms of the health of that axon, as I'll come on to in just a moment. So, in the absence of the myelin sheath, the action potentials now proceed at the speed of a tortoise rather than the speed of a cheetah. Okay, so let's now get on to the second of the questions that I posed at the beginning of this talk. What do we mean by the term myelin repair, or more accurately, remyelination? So, when oligodendrocytes are lost in the central nervous system, as occurs in an acute episode of the disease that we're discussing today, there is a spontaneous regenerative process that takes place. So the central nervous system, in all adult mammals, including adult humans, has the capacity to regenerate new oligodendrocytes. And this is a very positive and optimistic thing, because it contrasts very sharply with the consequences of the loss of the nerve cell. So if you lose the nerve cells, the 3 cells that you see over on the left, as is the case in the classic neurodegenerative diseases such as Parkinson's or Alzheimer's or Huntington's disease, the adult human brain has pretty much no capacity to generate new neurons. So if you lose neurons, they're gone forever. However, if you lose oligodendrocytes, you can regenerate oligodendrocytes with some efficiency, subject to various variables that I'll come on to shortly. So the regeneration of new oligodendrocytes is the process of myelin repair or remyelination. And those newly generated oligodendrocytes put new myelin sheaths around the nerve fiber, and so the whole architecture of the tissue is restored to what it was originally. So this is a true regenerative process. And this is really why I actually prefer the term remyelination than myelin repair. Myelin repair gives some sort of sense that there's holes in the myelin which you patch up. That's not what happens at all. The myelin sheath disappears in its entirety and it is replaced in its entirety. So it's a true regenerative process rather than a patching up repair process. So how does remyelination take place? How is it that the brain is able to regenerate new oligodendrocytes in the face of their loss? So now here's a somewhat more complicated diagram of the key constituents of the central nervous system. So running left to right are the green lines which constitute the axons or nerve fibers, and they'll be attached to a neuronal cell body at some point over to the left or right. And those nerve fibers are myelinated, as we've already discussed, by oligodendrocytes, which are the yellow cells that you see with processes going out top and bottom, attaching to the myelin sheaths that are surrounding the nerve fibers. Now, the other cells that I've included in this diagram are the blue cells with little processes sticking out to left and right. And these are the stem cells of the adult brain. Technically, we often call these cells oligodendrocyte progenitor cells, or OPCs, but they are, in effect, the stem cells of the adult brain. And it turns out that the adult human brain, the adult mammal brain, is full of these stem cells. They are a very abundant population. Probably about 6 to 8% of all the cells in your brain are these brain stem cells, widely and evenly distributed throughout the whole of the brain and spinal cord. That's most easily seen if you look in a laboratory species like a rodent, a rat, or actually in the image I'm just going to show you a mouse, where we can actually identify individual brain stem cells. So what you see here is a section through the brain of an adult mouse, cut in this direction, so we're going through the middle of the brain. And the reason this image looks slightly purple, and if you're in the front few rows you might see that there are individual dots, is because each of those individual dots that you see within this adult mouse brain is a brain stem cell. So you can see that they're very abundant, widely distributed throughout the whole of the mouse brain, and the same would be true if you followed on down the spinal cord, and the same would be true that if you looked in a human brain. So these stem cells are very abundant, and that's an important point for reasons, again, I will come on to shortly. So, how do these brain stem cells contribute to the process of remyelination? So let's imagine that within this area of tissue, where we have myelinated axons running left to right, an episode of demyelination takes place. So demyelination is the process which occurs when the oligodendrocytes are killed and the myelin sheaths come off the axon, leaving them demyelinated. So we've killed the oligodendrocytes, and as a result of killing the oligodendrocytes, the myelin sheaths degenerate, and the axons become demyelated and exposed. And of course, as I've already mentioned, that'll have immediate consequences for the ability of that nerve fiber to conduct electrical impulses efficiently. Now, in response to that damage, that injury occurring in the brain and spinal cord, the whole milieu of that tissue changes in such a way that the stem cells that are present in the vicinity of the injury are stimulated to respond. So the first thing that happens after you lose oligodendrocytes is that the brain stem cells around the area of damage recognize that something has gone wrong. They change their whole state of being and start to make copies of themselves. In other words, they proliferate, and they also start to move around, they migrate. And so what happens is that that area of damage fills up with recruited brain stem cells. So they respond to signals that are generated by the damaged tissue. They become activated. They don't actually throb like this, but that's just illustrative of their change in state. And in that changed state, they make copies of themselves that move around, and they fill up the area of damage. And that's the recruitment phase of remyelination. So very quickly after you lose oligodendrocytes, the area of damage is filled up with brain stem cells. This is an illustration from an experimental animal where we've been able to genetically label the stem cells so that they fluoresce green. And this is a very, very small component of the spinal cord of an adult mouse where we've killed the oligodendrocytes. And shortly after the loss of the oligodendrocytes, it's such a small area that the mouse is completely oblivious to the fact that we've killed a few oligodendrocytes at some point in its spinal cord. And shortly after the loss of those oligodendrocytes, that area of damage fills up with these stem cells. So you see this area of damage now chock-a-block full of green stem cells. And these sort of models are extremely useful to us as experimental scientists studying the process of remyelination because we can now see the stem cells and we can study their behavior in some detail and the factors which regulate and control their behavior. So the final phase of the regenerative process of remyelination is when the recruited stem cells become the missing cells. And the missing cells here are, of course, the oligodendrocyte. The process of a stem cell becoming a very specialized type of cell, such as an oligodendrocyte, is the process that we call in stem cell biology differentiation. So the second and final and critically important phase of remyelination is when the recruited stem cells become oligodendrocytes that grab hold of the demyelinated axons and reinvest them with new myelin sheaths. And at the end of that process, again, the architecture of the tissue is restored to normality. So the axons are now surrounded by myelin sheaths, which will allow them to conduct by this rapid mechanism of saltatory conduction, the speedy conduction of impulses along the length of the nerve fiber, and will also help protect the nerve fiber. So that, in a nutshell, is how remyelination takes place. Of course, the mechanisms driving this are very much more complicated than I've conveyed in these diagrams, and we'll come on to some of those mechanisms shortly. So the third question is, why is myelin repair such an important process? And given what I've said so far about the function of myelin, you can begin to anticipate what the answer to that question might be. So let me just step back from this and give you some important context to why myelin repair is such an important process. So represented here is the inescapable process which affects all of us of aging. We are all aging all the time. So we're now older than we were when I started this talk, and you may feel that you're much older than you were when I started this talk. So multiple sclerosis is a long-term disease. Commonly, it occurs in individuals in early adulthood. And as you know, the life expectancy of people with the disease is not shortened by that very much compared to the normal population. So this is a disease that people will live with for several decades. So this is a disease that people commonly have throughout their adult life. So the process of aging is likely to be pertinent to how the disease unfolds as you move through adult life. And it transpires, and is perhaps not surprising, that perhaps the most important determinant of any regenerative process in any tissue is the process of aging. So as we get older, our ability to regenerate tissue decreases. And in some sense, that's why we age, because we're not able to regenerate tissues as efficiently as we were when we were younger. This is illustrated in this first data slide of my presentation, which is taken from a paper published over 40 years ago. And it's a classic paper which looked at the effects of aging on a very simple regenerative process of wound healing. So this is a breach in the skin, a wound that then has to heal. And between the ages of 20 and 40, the rate of healing of that wound is halved. So it takes twice as long to heal a skin wound between the ages of 20 and 40. And this is a progressive process. So if you looked again at 60 and 80 and so forth, the rate of wound healing would be decreasing as you age. So this is really illustrative of this very important concept that aging affects the efficiency of all regenerative processes. And it turns out that you could replace any tissue you like here, and the phenomenon would be exactly the same. So you could put in liver, you could put in the bone marrow, you could put in any tissue you like, you could put in bone here, and it would be profoundly affected by aging. So here is some other diagrams illustrating this concept of the effects of aging on regeneration, looking at the ability of bone to regenerate. This is a young adult rat of about 3 months of age, and an experimental fracture has been made at this point here. And 80 days after fracture, it's almost impossible to tell where that fracture took place because the bone is healed completely. But in an old adult rat that's about 24 months of age, at 80 days, it's still very clear, if you peer closely, but it is pretty clear, that bone is still not healed. And that, of course, is why skiing is a pretty reasonable sport for somebody in their early adult life to engage in, because if they break their leg, it's going to heal pretty quickly. But if you're in your 60s, 70s, and 80s and you go skiing and you break your leg, then it's going to take a lot longer to heal. So this phenomenon of aging affecting the efficiency of regenerative processes occurs in all tissues in the body and absolutely applies to the efficiency of regeneration of myelin sheaths. So as we get older, as we progress through adult life, with the disease, our ability to regenerate new oligodendrocytes is declining simply as a function of the clock ticking. It's got stuck. Here we go. Okay, so the implications of that is that early on in the disease, once this process has occurred, so you have an episode of inflammatory demyelination taking place at some point in the central nervous system, if you're a young adult, then the chances are you'll regenerate oligodendrocytes pretty quickly and pretty efficiently. But as you progress through the disease, as you age, the efficiency of this process is declining, and it can become so slow that, to all intents and purposes, it appears to have failed. So remyelination could become so inefficient that de facto, the remyelination has more or less ground to a halt. Now, if demyelination occurs and there is no regeneration of the myelin sheath, the axon is then left exposed for longer than is good for it. So not only does that axon now lose the ability to conduct electrical impulses efficiently, but because of the second function of the myelin sheath, of maintaining the health of the underlying axon, in the absence of the myelin sheath, that axon is very vulnerable and liable to undergo degeneration itself. And as I mentioned earlier, if you lose axons, nerve fibers in the central nervous system, there is precious little by way of spontaneous regeneration. So this is an irreversible change. And sometimes, in fact, the whole nerve cell can die, not just the nerve fiber, but the whole nerve cell. And again, as I mentioned earlier, if you lose nerve cells, they're not spontaneously regenerated. So if you get to this point where there is no restitution of the protective myelin sheath, the nerve fiber will degenerate, and because that's irreversible, the accumulation of loss of nerve fibers leads to the progression of the disease. And so what is really happening in the secondary progressive phase of the disease, in fact, it's happening already in the early relapsing remitting phase, but certainly in the progressive phase of the disease, there is the driver of that is the cumulative loss of these irreplaceable neuronal elements of the central nervous system. And that's why putting myelin sheaths back on axons is a critically important way of slowing or arresting the progressive phase of disease. And our efforts in Cambridge and Edinburgh over the last 8 years or so have really been focused on this ultimate endpoint of coming up with some strategy for trying to address the, at present, untreatable progressive phase of MS. So clearly, there is an important therapeutic, urgent, I would say, need for making sure that when this process occurs in the central nervous system, you always go down the regenerative protective route of reconstituting the tissue and putting the protective myelin sheath back on the nerve fiber, and avoiding going down this degenerative route where remyelination has failed, and degenerating because remyelination has failed. So let's now think about how a regenerative therapy fits into a broad strategy for treating multiple sclerosis. Now, clearly, if one was thinking about this as a conceptual thought experiment, the first thing you'd want to do is to try and stop the damage occurring in the first instance. Clearly, you'd want to try and intervene at this point here to stop demyelination taking place. Now, as I've mentioned to you, and you'll have probably heard from others on many occasions, the demyelination is occurring because the immune system goes awry and starts to kill oligodendrocytes and damage the myelin sheath. So clearly something that changes or modulates or suppresses the immune system will help in preventing this demyelination occurring in the first instance. So the first strategy of stopping damage is the whole area of immunomodulation, the immunotherapies. And pretty well everything that is currently available that you may be on or may want to be on or might be on at some point in the future, all of the drugs that are currently available, the disease-modifying drugs, are really aimed at modulating the immune system such as to stop the damage occurring in the first instance. Now, ultimately, it may well be the case that those therapy interventions are so effective that we can completely shut down the disease from the outset. And that, of course, would be a very that would be an objective that one would hope to reach at some point in the future. But here and now, it's naive to think that we're going to be able to completely suppress that damage from taking place. So it will be the case that some of the some disease process will escape the disease-modifying interventions and demyelination will take place. And at this point, you really want to have a regenerative therapy, which makes sure that you go down the regenerative pathway and not down the degenerative pathway. And by implication, what I said earlier, the further you on in the disease, the greater the urgency and the need for regenerative therapy, because your own natural healing processes will be becoming tired as a result of aging. So this has really been the focus of the two centres in Cambridge and Edinburgh over a number of years, to see whether we can come up with ways by which you can promote myelin regeneration in those situations where we haven't been able to completely suppress the disease process by the currently available disease-modifying drugs. And this is now, I think it's true to say we were sort of pioneers in this area and have been for a number of years. And the MS Society, of course, has been, in that sense, also a pioneer in their investments in Cambridge and Edinburgh, but is now, I think, universally recognized internationally as the major urgent objective in new therapies, new radical therapies for this disease. So then the last question, how can we do this? What approaches can we take to try and make sure that we put myelin sheaths back on axons after demyelination has taken place? Well, the classical way of thinking of regenerative medicines, the approach that you read about in the daily press is the concept of cell transplantation. For many people, this is the top of the list of the ways that you can regenerate tissue. You transplant a cell into the damaged area, and that transplanted cell replaces the cells that are missing, and you're back to where you were, healed. And for many people, the best cell to transplant would be a stem cell. With the proviso that I'll add, that the term stem cell actually embraces a whole range of different cell types. There is not a single entity of a stem cell. So one has to be very wary when you read reports of the efficacy of stem cells in the paper. Firstly, whether it's true or not, but secondly, precisely what type of stem cell is being talked about. There is not a single entity of a stem cell. But it would not be unreasonable to think that in a demyelinating disease, you might be able to transplant brain stem cells that we know are capable of making the oligodendrocytes into an area of demyelination in the brain, and those transplanted cells would replace the missing oligodendrocytes. Well, that conceptually is a very straightforward approach to myelin regeneration. Well, the first thing is, where are we going to get these cells from? So that's not a trivial question, by the way. One possibility is that you could actually grow these cells from the adult brain. So you could take a bit of brain from a patient, identify these cells, grow them in tissue culture, make lots of them, and then transplant them back into the brain. And given what I said to you earlier, that there are an awful lot of these cells in the brain, on the face of it, that seems not to be an unreasonable approach. And it seemed to be a not unreasonable approach when we first entertained this idea about 20 years ago. Transpires that it's extraordinarily difficult to grow these progenitor cells from the adult brain for reasons that are still not really understood or been addressed. In the last few years, alternative ways of growing stem cells have emerged. So you may have come across the concept of the iPS stem cell. This is the work that Yamanaka and Gurdon won the Nobel Prize for last year. And this is the idea that you can take any cell in the body, so you could take some cells from the skin, and that's a very trivial way of obtaining cells, and by a relatively short number of steps, in fact, a surprisingly short number of steps, you can make any cell in the body into an embryonic stem cell. Embryonic stem cell has the wherewithal of become every cell in the body, and so you can go from the embryonic stem cell towards the cell that you want. So you can take any cell in the body, make into embryonic stem cell, and then differentiate that embryonic stem cell into, for example, a brain stem cell that has the capability of becoming oligodendrocyte. And that technology is rapidly becoming available. So there are now a number of publications where people have described methodologies for obtaining human oligodendrocyte or brain stem cells from something as trivial as a skin cell. But there is a second problem in addition to where you would obtain the cells from, and that is the feature of multiple sclerosis, in that this is not a disease where there is a single area of damage in the brain that occurs as a one-off event. Instead, as we know, the damage within the MS brain occurs in many points at different times. So transplantation for multiple sclerosis would be very different from cell therapy approaches, for example, to spinal cord injury, which some of you might have come across in the press, where you have a single episode of damage occurring at a single point in the spinal cord. So you put cells in there, and that's relatively straightforward. Same would be true for Parkinson's. There's a single area within the brain where you lose neurons, and so you transplant cells into that single area within the brain. That is not true in multiple sclerosis. The damage is occurring at different points throughout the brain and spinal cord. And so that makes it a challenge to think how you would transplant into all of those areas at different times during the disease course. And that is not a trivial challenge by any stretch of the imagination. And it is largely for that reason, actually, that many of us have become more attracted to a second approach to promoting remyelination in the MS brain. But before I get onto that, I just want to clarify something which I think causes a lot of confusion in the community. So there are stem cell therapies available at present and being trialed in various countries for multiple sclerosis. In all instances, I think I'm right in saying, the stem cells that have been used here are stem cells obtained from the bone marrow. Very often they're a type of stem cell called a mesenchymal stem cell. Not always, they're sometimes hematopoietic stem cells, but generically bone marrow stem cells. Now, I think it's very important to understand, because there is this sort of zeitgeist and popular conception at the moment that stem cell transplantation is always a regenerative strategy. That is not the case in MS when you transplant bone marrow stem cells. So bone marrow stem cells are not directly regenerative in MS. I thought that was my cue to shut up, but right, so let me just explain that now in the context of the two approaches that I described to you for treating MS. So you remember I said there were two approaches. One is to stop the damage from taking place, and that's to modulate the immune system, and the other is to promote the regenerative process. Bone marrow stem cell transplantation is really targeting this. It's stopping the immune it's modulating the immune system, which is causing the damage. So that we know that bone marrow stem cells, and particularly mesenchymal stem cells, are very efficient at changing immune systems, changing the immune response. Exactly how they do that is still a bit of a mystery, but we know they're very efficient at doing that. So bone marrow stem cell transplantation is not a regenerative therapy, it's an immunomodulatory therapy. So it is in the same category of treatment as all of the drugs that you know that are currently prescribed and used for the treatment of the disease. They are not directly regenerative therapies. So you do not go to get a bone marrow stem cell transplantation therapy with a view directly to being regenerated. That's not how bone marrow stem cells are working, and I hope that's clear. It may be the case that in modulating the immune system, they change the environment in such a way that this remyelination process is more likely to take place. And that's an intriguing possibility for which some evidence exists. But it is not the case that these bone marrow stem cells will generate new oligodendrocytes directly as a regenerative process. Okay, so that's really a summary of transplantation approaches to curing or remyelinating in multiple sclerosis. Brain stem cells will make oligodendrocytes, but how you get them to all the different areas in the brain where they're needed is a difficult challenge. And bone marrow stem cells, when they're transplanted, are not directly regenerative. That's why some of us have been entertaining this alternative approach to promoting remyelination. That is based on the fact, which I've already introduced to you, that the brain is full of stem cells that are capable of becoming oligodendrocytes. So we know that these stem cells can become oligodendrocytes. And we know that they do that very efficiently in young adults. And we also showed in a paper that received some attention about 80 or so months ago that even the aging stem cell is still capable of making an oligodendrocyte. So it ought to be the case that you can do something to the stem cells that are already present in your brain to make them regenerate oligodendrocytes more efficiently. So instead of transplanting stem cells, you target the stem cells that are already present in the brain. This is sometimes called the endogenous approach to remyelination because you're targeting the endogenous stem cells already present in the brain. And that is going to be, in all probability, a pharmacological approach. Instead of a transplantation of cell approach, instead a drug which somehow changes the behavior of these stem cells, making them become oligodendrocytes more efficiently. So this slide here just summarizes the conceptual approach that we've taken over a number of years to try and see whether we can come up with drugs that make stem cells, make oligodendrocytes more efficiently, and would therefore be the basis of regenerative medicines. Now, if your car breaks down, you are in no position to fix it unless you understand how the engine works. So a priori to fixing a broken car is you have to understand how the engine works. And if you understand how the engine works, then you can figure out what's gone wrong when it breaks down. And if you know what's gone wrong when it breaks down, then you can repair the car. So all people that come out to fix your car understand how the engine works. I'm certainly not in that category. But that's pretty much the approach that we have decided to adopt in coming up with ways of promoting or fixing myelin regeneration, myelin repair. First of all, we have to understand how remyelination works when it works efficiently. So how does the car engine work when it works normally? How does remyelination work when it works efficiently? If we understand that process, we can understand why it goes wrong, and if we understand why it goes wrong, then we can fix it. And that is why much of the time has been spent, much of the investment has been spent from the MS Society in really trying to get to grips with the fundamental biology of how remyelination takes place. So the diagrams that I showed you earlier on showing how remyelination occurs, where you get stem cells that get recruit and differentiate, that was crucial information to understanding this process. And a lot of that understanding has come from work that we've done with MS Society investment. Now, it turns out that as you get older, the stage of remyelination which is most seriously impaired by the process of aging is not the abundance of stem cells. The number of stem cells in your brain stays about the same regardless of your age, nor is it their ability to be recruited, but it's the final stage of remyelination where the recruited stem cells have to become new oligodendrocytes. So commonly, if you look in areas of the MS brain where remyelination hasn't taken place, it looks like this. So you see demyelated axons, and you see plenty of these stem cells, but they haven't yet been able to become oligodendrocytes. And it's for that reason that we've spent a lot of time in the two centers in trying to understand the mechanisms, the machinery by which a stem cell is able to engage an axon, become an oligodendrocyte. So what are the intrinsic and extrinsic processes, in other words, the processes within the cell and the processes around the cell that are making that stem cell become an oligodendrocyte? This turns out to be an extraordinarily complicated bit of biology. There are multiple mechanisms working interactively with each other in different degrees of importance. But we have been, I think, rather successful over the last few years, again as a result of the investment the MS Society have made, in identifying a whole host of critical processes which are involved in stem cells becoming oligodendrocytes, and those are listed here. Needless to say, these processes are related to pathways or molecules and therefore have the somewhat abstruse names that typify biological science. But these pathways or molecules are listed on the left here. They've, for the most part, well, pretty much always been published in very high-impact journals. And some of these processes or pathways or molecules are processes which make stem cells become an oligodendrocyte, and some of them are processes which stop cells becoming an oligodendrocyte. And if they're responsible for making a stem cell become an oligodendrocyte, they're blue, and if they're responsible for stopping a stem cell becoming an oligodendrocyte, they're in red. So if it's a process which makes a stem cell become an oligodendrocyte, then you design a drug which replicates that. If it's a process which stops a stem cell becoming an oligodendrocyte, you design a drug which inhibits that inhibitory. So two inhibitions cause a positive. So this first target was the first one we published. This was a collaborative work between my lab in Cambridge and Charles French-Constant's lab in Edinburgh. The Wnt pathway was a collaboration between my lab in Cambridge and some colleagues at University of California, San Francisco. Endothelin-2 and activin-A were both targets or mechanisms which were joint ventures between Cambridge and Edinburgh. PDE4 was a collaboration between my lab and a colleague in Cambridge, Mark Cottrell, I'll mention in just a moment. And this is a paper which is going to come out shortly where we've identified another mechanism by which you can promote remyelination, which has been done in collaboration, again, with colleagues at University of California, San Francisco. Let me just highlight two of these that have been published within the last 6 months. The first of these is a molecule called activin-A, which actually turns out is made by inflammatory cells which arrive in areas of damage. So it turns out that not all inflammatory cells are necessarily bad. There are types of inflammatory cell which turn out to be quite good and rather important for regeneration to take place. And those good inflammatory cells produce a range of molecules which help the regeneration of myelin take place. And one of those molecules is activin. And this was discovered by Veronica Miron, who is a postdoc working jointly between my lab and Charles's lab, and now is running her own laboratory at the University of Edinburgh. This is very early days in translating this particular discovery into a therapy. There's still a long road before that takes place. But it's very important to have identified these starting points in that sort of translational process. And the second of these, the phosphodiesterase inhibitors, this was work that was led by Mark Cotter, who was an ex-student of mine, who now runs his own laboratory in Cambridge, and we published this jointly last year. And these are interesting, exciting stories which will unfold over the next few years, and we hope eventually lead to new therapies. Now, there is a pipeline, a process from going from a scientific discovery in the laboratory to a clinical intervention. And that pipeline can be long or short, depending on what's available, what's known, what the type of discovery is in the first instance. So the first of the discoveries that we made and published a few years ago was this compound here, RXR gamma. And we're just going to tell you a little bit more about that, because that is the one that is furthest along this pipeline from being a laboratory discovery to a clinical intervention. So RXR gamma is a very interesting molecule that is rather complicated. Part of the molecule can receive drugs and compounds coming into it and can attach to that molecule, so it acts as a receptor. And another part of the molecule will actually engage in the nucleus and change the whole behavior of the cell. So these are really rather interesting and important molecules, and I can tell you a lot more about those should you wish, but that's the essence of what these molecules do. So RXR gamma is expressed within brain stem cells, and if you activate this molecule, the stem cell becomes an oligodendrocyte much more efficiently. So what we showed in the paper we published a few years back was if you take old rats, so these are old rats that when you demyelinate them, they're going to remyelinate very slowly, and you give those rats a drug or a compound which activates that molecule RXR gamma, you can make the remyelination much more efficient. And what you have to grasp in here, in this figure here, is that if it's a pink circle, the nerve fiber's been regenerated with new myelin. If it's a gray circle, it hasn't. So this is the control old animal, which has just been given saline, salt water. Very few of the axons have new myelin sheaths and are pink. We've made them pink. That's sort of they're not actually pink. Very few of them are pink, but if you give a molecule which activates this RXR receptor, then many more of them are remyelinated. And this actually was quite a dramatic discovery. So this was actually the first time we, or anybody for that matter, had made an old animal make myelin sheaths more efficiently by giving a single molecule. So you can see here very clearly that we've promoted remyelination. There are more pink fibers on the right than there are on the left. So RXR gamma is a very important target for making stem cells into oligodendrocytes. Are there any drugs which target this receptor that might be available as a regenerative medicine in MS? Well, as I said, the rate at which you move along the pipeline varies from what's available and what's known and what the target is and so forth. Actually, for the RXRs, we can sort of fast forward along the pipeline reasonably quickly because it transpires that there is already in existence a drug developed and licensed for a completely different indication which targets this particular receptor, and that is a drug called bexarotene. So, bexarotene activates RXR much in the same way as the molecule that we used experimentally in the previous slide I showed you. So would it be possible to use bexarotene as a regenerative medicine in MS to make the brain stem cells make oligodendrocytes more efficiently? Well, we are in the process of planning and setting up a very small toe-in-the-water clinical trial using bexarotene as a regenerative medicine in MS. Now, this is an area where I have to hand over as an experimental biologist to my clinical colleagues who understand running drug trials and all the gamut of issues associated with that. So this particular trial, which is in large part funded by the MS Society, will be run by my colleague in Cambridge, Dr. Alistair Coles. So Alistair Coles has been engaged in MS clinical trials for many years, is one of the world's experts in this. In fact, Alistair is the one who ran the alemtuzumab trial. So Alistair will be leading this clinical trial, and it'll be a collaboration with Siddharthan Chandran at the University of Edinburgh, and also David Miller, who runs the MS Society-funded imaging centre at the Institute of Neurology in London. So the trial is what is called a randomized placebo-controlled study, and it is addressing the safety and tolerability of bexarotene, an RXR receptor agonist, with the specific objective of testing its ability to promote remyelination. And this particular trial, for reasons I'm happy to discuss later, and as much as I understand it, as I say, I'm not a clinician, will be done in individuals with relapsing remitting MS who are already on beta interferon therapy. Patient treatment recruitment will be in the 2 centres in Cambridge and Edinburgh. The outcomes will be on the basis of MRI scans, which will be done in London, Edinburgh, and the analysis will be done by David Miller's group at London. Now, all being well, the details, the crossing of the T's and dotting of the I's of the funding of this is still under negotiation. But all being well, I am told by Alistair that he's hoping to start recruitment in the autumn of this year, so autumn 2014. A little bit more details on what this trial will involve. Technically, it's called an add-on placebo-controlled trial. Add-on in that individuals will already be on a treatment, beta interferon, and placebo-controlled in that some people will get the drug and some people will get something that looks identical to the drug but isn't, and nobody will know who's got what. Bexarotene will be given in tablet form on a daily basis, and the participants in this first, as they say, toe-in-the-water trial will be individuals with relapsing remitting disease with an EDSS of between 3 and 6, between the ages of 30 and 50. This is a very small-scale trial in the first instance. This is really virgin territory. There is one previous clinical trial with a putative pro-remyelination therapy that has been done anywhere in the world. So this is very early stages. And this initial trial will involve 50 patients, 25 who will get the drug, 25 who will get the placebo. The trial will run over 9 months, 6 months of which will be on treatment. So that's a very optimistic and a very encouraging outcome of the investment in fundamental science. And I think it's worth reiterating here that unless there were the investment by the society in doing the fundamental biological science of identifying this interesting molecule, we'd have never got to the point of being able to do a clinical trial. So I think it illustrates the importance of funding fundamental science for translation. I'm just going to finish on telling you where we're moving with this. It's slightly more complicated than I've told you, and things always are. RXR actually comes in 3 different flavors, an alpha, beta, and a gamma form. The gamma form is the one that is found in the brain. The other types of RXR are found in other tissues in the body. Bexarotene is actually targeting all 3 types of RXR. So although it'll affect the brain stem cells, it'll also affect cells in other tissues. So there will be some what are called off-target or side effects of using bexarotene. They're not expected to be terribly serious and they're expected to be manageable. Nevertheless, there will be some off-target effects. But clearly, it would be much more optimal if we could come up with a drug that was specific to the gamma form of RXR. So it would have optimal and maximal effect on brain stem cells and not affect the other tissue. And that is likely to be a more effective regenerative medicine than bexarotene. So bexarotene we have here and now, and it is absolutely sensible and worth trialing that at this stage. But we're also engaged in what's called a drug development program, where we're going to try and devise new drugs which are specific to the type of RXR that is found in the brain. And that's a much more long-term process. It'll take between 5 and 15 years. So let me just summarize what I've said. Promoting remyelination is important in MS because it'll help restore function, and perhaps even more importantly, it'll stop the nerve fiber loss that is the basis of the progressive phase of the disease. We think in Cambridge and Edinburgh that currently the best way to enhance remyelination is not by cell transplantation, but instead to design drugs which target the stem cells that are already present in the brain. And we've got pretty close to doing that. We're on the brink of trialing just such an approach. The two centres working jointly and with other collaborators in the international community, we've now identified a number of possible ways by which we can come up with regenerative medicines. These are at different stages of development, some at the very early stage, some much more advanced. The most advanced of these is the whole approach around RXR gamma and the bexarotene trial, which will start during the course of this calendar year, all being well. And that I think is where I wanted to conclude, just to emphasize again that we have made, with the help of the investment of the Society, really, I think, better progress than I would have expected when we embarked on this whole venture. When we first started to engage with the MS Society in the notion of setting up a myelin repair center, the idea that we'd have medicines to promote myelin regeneration was really aspirational. And I think as a result of being very fortunate to employ some extremely talented young scientists on MS Society money, we've made much more rapid progress than we might have expected. And I think the notion of regenerative medicines is now a realistic one. And I, for one, am very optimistic about how the next 5 to 10 years is going to unfold. Thank you for your attention. I'm happy to answer questions.
Fantastic. Thank you, Robin. Now, officially, we've actually used our time, but I've been given permission to extend a bit so we can have some questions. So if there are some questions, that would be great. And we have one right down at the front in the left.
Very good talk. Thanks, Robin. If remyelination and demyelination, or demyelination and remyelination reflect the relapsing remitting forms of MS, what do you think is happening in the progressive forms when there's no relapsing and remitting phase?
Well, I think that's a very good question, of course. The whole community is trying to get some clarity on. But I mean, there are many components to that answer, but I think it's not unreasonable to think that when you go into the second progressive phase, what's kind of interesting is that people enter the secondary progressive phase at around the same age, regardless of the age of onset of the disease. So this is work from Christian Confrèvre in France that he published a number of years ago, and it's roughly around the age of 40, regardless of when the disease started. And that, to my mind, speaks very eloquently to the idea that what is happening when you enter the secondary progressive phase is you reach an age where the efficiency of remyelination, simply by virtue of your age, has now become so slow that the axons are left naked for longer than is good for them. And when you go into the secondary progressive, you've now entered a phase where actually the new episodes of disease is diminishing, but the axons are remaining exposed, therefore not receiving their nutrient fix from the myelin sheath and undergoing degeneration. And that's an irreversible loss, and the more of those nerve fibers you lose, the more disability you get, so it becomes a progressive disease. And that's how I conceptualize it at the moment.
And another question with Nicola.
Yes, thank you. Thank you, Chairman, and thank you to the speaker. Very clear, I thought. I'm not a scientist, but I thought for a non-scientist, extremely clear. The focus clearly is on repair. I'm just wondering what your work shows and what it helps us on repairing and stopping the damage in the first place.
Well, so these are two separate areas of research. I mean, neither our centre or Edinburgh Centre are really engaged in that whole process of stopping damage, and that, I would say, is probably accounts for 98% plus of the investment in disease treatment interventions over the last 20 or 30 years. So our particular interest has been, I mean, our focus is on repair, not the exclusion of those other therapies. And in fact, actually, the success of those therapies is really the basis on which a regenerative medicine is going to work, because clearly, unless you can stop the disease process, then you can regenerate until you're blue in the face and you're still going to have damage. So these two interventions work sequentially with one another.
Okay, 2 more questions, I think, and then we'll conclude. And I've got one with Sue right at the back, and then one towards the front with Nicola.
Yeah, can I ask, when you talk about the myelin regeneration, so if that becomes what you want it to be, for people who have had the disease for a long time and have entered into that phase where they've had demyelination and nerve loss, regeneration is not going to help them. Is that right, from what I'm reading from what you're saying?
Yeah, that's a very good and very important question. So remyelination clearly won't restore lost nerve fibers, and you can't remyelinate a fiber that isn't there. But at any stage of the disease, you still want to protect what nerve fibers you have. So if one is being very pessimistic about this, then the remyelination therapy will slow the progression. If you're being more optimistic, remyelination therapy will stop the progression. If you're being very optimistic, and in fact there is sufficient biological knowledge to think that this may be the case, if you stop progression of the disease, there may be sufficient what's called plasticity in the brain to actually recover some function. So if you stabilize things, the brain actually is remarkably good at finding new ways of doing old functions that it had lost the ability to do. So it may well be the case that remyelination therapies will lead to a recovery of function by stopping the disease process and allowing the brain to engage in these alternative routes of doing the same function.
Down at the front.
Well, one of the great advantages of medicine in dealing with diseases over the years has been vaccination. Vaccination stimulates the immune system of the body to deal with invasive and problematic things. Do you think the vaccination over the years has been some kind of a cause for the stimulation of the immune system that's affected MS?
Yeah, so this is a good question. Has been investigated pretty thoroughly by immunologists or people who have better immunology knowledge than I have. And it's been one of many things, one of many potential triggers for the disease. And the evidence thus far is there is no compelling evidence thus far that vaccination has been the cause of tipping the immune system into this aberrant form of behavior. So while it's an interesting concept, there's no evidence that that's the case.
Brilliant. So I'm going to make a couple of thank yous, but before I do, I just want to remind people that we do have a science zone right at the back of the hall where you can learn a lot more about myelination, demyelination, and oligodendrocytes and various other things connected to the central nervous system. But my 2 thank yous, the first is to all of you as people who are part of the MS community that help the MS Society to be able to fund the kind of work that you've heard about today. That's incredibly valuable. And the second, of course, is to Robin Franklin for giving up his Sunday, for coming to talk to us, and for all the work you do day in, day out to make the difference we want you to make. Thank you.
Could I just ask people if they could leave the theatre via the back, not through the sides? Thank you.
And we'll be back here in half an hour for another session on stem cell research from another angle. Thank you.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Live experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Live, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Live that you can't see, and I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
I stand tall, watching everything that life gives to
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Live experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Live, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Live that you can't see, and I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Live experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Live, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Live that you can't see, and I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Live experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Live, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014. I'm with Shaa Pizaro, who is going to be doing a talk, Escaping Labels. Hello, Shaa.
Hello.
Could you tell us a little bit about the talk and what you're going to be talking to people about?
Well, I'm going to be talking about a postcard that I entered into a project called Postcards from the Edges. And the brief was, what would you want to say to the world about being disabled? Put it on a postcard. And I sort of laughed, and there was a picture of me smoking in a back alley, and I wrote, I drink, I smoke, I swear, on it, sorry, and sent it off. It got a lot of press attention, and it's been, travelling around the country in an exhibition and ended up in Parliament. Fortunately, I know a lot of politicians quite well, and just said, oh, it's what you'd expect from me, and they laughed. So, so I'm talking about that, and I'm talking about why I did that postcard, and actually what's behind it in terms of dealing with a diagnosis with MS, coming to terms with being disabled, and the way that MS affects my life, and the way that I've kind of overcome that to make sure that I still get to be me.
Now, the talk you're doing is Escaping Labels. Could you talk to me a little bit about what labels you're thinking of escaping?
Well, I talk a bit about how disabled people are represented in the media, because I think that very much there's sort of three categories that the media present disabled people in, and one is Paralympian supergods, the second is liars and scroungers and benefit cheats, and the third is sort of tragic cases that need to be pitted, and I don't think I'm any of them. And I don't think most disabled people are any of those. And so, I want to talk a bit about that.
Also in the talk, it's talking about low self-confidence. Could you give some tips about low self-confidence and how to overcome that?
I was in a marriage to someone who couldn't cope at all with the diagnosis of MS, and he very vocally would tell me that I was disgusting because I was disabled and that he couldn't find me sexually attractive, knowing that I had to catheterize. You know, he was really very abusive, and in fact my divorce got put through as a priority case on the grounds of abuse. I knew that I couldn't handle that myself, and you know, I couldn't see how I could ever sort of regain my self-esteem and my self-confidence without getting professional help. And so I went off to a counsellor, and I found the counsellor that was right for me, and I spent 6 months with him. And at the start of it I said, I want to believe that I'm still me, and that I am sexy, and that I can, you know, still be found attractive. And through seeking that professional help, that was the thing that really changed things for me. So I think that would be my real advice to people, is seek out and accept the help that you're offered.
Thank you very much. And I think everyone could agree that Shana's looking very sexy today.
I'll take a couple of minutes while people continue to sit down, just so you know that we've erected that barrier because various people were tripping over the cable protector, which is designed to stop people tripping, but is probably the biggest cable protector I've ever seen. And so we've put a barrier up to prevent people from tripping over it. You heard a bit about stem cell therapy from Robin Franklin earlier, quite a bit in many ways. But one of the things that Robin talked about was the different kinds of stem cell therapy and the different ways that it's being deployed in MS. And so we now have Dr. Peter Connick to talk to us some more about some of those other ways that stem cell therapy is being used. Peter is a consultant neurologist at the University of Edinburgh. He works at the Anne Rowling Institute, I believe. And Peter is a really good advert for why it's important to fund PhDs, because Peter actually did his PhD in the use of stem cell therapies as a potential treatment for progressive MS, and that's still very much his area of expertise. So it's a really nice advert for why it's important. Peter wasn't actually funded by the MS Society for his PhD, but we fund many, and it's a lovely example of why it's important to do that, so we continue to grow the scientific community, which will take forward the research we need to see. So again, we'll have Peter talk to us, and then we will have more time for questions afterwards. Peter, thank you.
The talk, after that frisson of professional pride and excitement, I hurriedly replied, "Many thanks, of course." Those of you who are interested in how that building and that facility to do clinical research in regenerative medicine and regenerative neurology came about, J.K. Rowling is guest editor on Women's Hour tomorrow. and I believe she's going to spend some time there talking about it, so that may be of interest to you to tune into that. But as for the next half hour or so, what I thought I would talk about is really 4 areas, and I'll spend the most of the time talking about the first 2 because I feel really passionately that that's the key for what stem cells are. Robin touched on that. It's a bit of a black box, I suspect, to many people. They are more complicated than just a monolithic single entity, so we'll cover that in some detail. How might they be relevant to MS? And I've got a list of 5 areas that, if there were a sort of take-home assessment, test yourself, I guess that's what it would be from this talk. Can you remember the things that stem cells might do to be helpful in terms of therapies and developing our understanding of the disease? Current stem cell research, I'll perhaps spend a little bit of time talking about that most dangerous of subjects, which Nick has already mentioned, talking about what I did during my research in Cambridge. Those of you who have met PhDs in the past, never let them talk about what they did during their PhD research. You'll be here till tomorrow, frankly, so I'll try and be brief. And the future of stem cell research in MS I'll touch on, because actually I think the key is for you to tell us what the future of stem cell research is, rather than us to tell you. So what are stem cells? Fundamentally, these are a type of cell within your body, and there are about 260 plus different types of cell within you, which have 2 properties. Normally, when cells divide, they produce 2 copies of themselves. Stem cells are different in the sense that they produce 1 copy of themselves and 1 copy that is either specialized or has the potential to become specialized. So they're able to self-renew, they're able to create a copy of themselves, and they are also able to create something that's a bit more specialized, or at least could end up that way in time. That property is called asymmetric division. And every tissue you have pretty much has stem cells within it. So you have them in your bone marrow, where they're responsible for making bone, fat, cartilage, bone tissue. You have them in your bone marrow where they're responsible for making blood, red blood cells, white blood cells, and platelets. You have them in skin, gut, etc., etc. And Robin used liver as a good example. We have them in our brain, as you heard earlier, and part of the interest is whether those are a relevant population or whether other non-brain stem cells are relevant to MS, and if so, how might they be relevant? Now, not all stem cells are the same. That's an important thing to start with. Now, I had hoped to give you an image, but please don't, at least not if you're at work, ever Google image search potency. The key property really is what types of specialized cells can a stem cell make? I'll pause there and just let that one sink in. When you're an adult, and I'll come on and describe this again just so that it really resonates, when you're an adult, you tend to have tissue-specific cells, which are stem cells, but which are only really able to make the kind of cells relevant to the tissue they are in. For example, in the bone marrow, be able to make bone marrow type cells. In the liver, be able to make liver type cells, for example. The jargon of this is encapsulated by that little list of 4 things there, and I'll run through them. Totipotent we almost never hear about, and you probably haven't come across it before. Just after fertilization of the egg, in that first few days of life, our cells are able to make all of the embryonic tissues, so all of the tissues that become a human, and all of the extra embryonic tissues, the yolk sac, etc. That's rapidly changed to a pluripotent state, and these are the exciting embryonic stem cells that you've probably heard and read about, cause such controversy around the ethical justification for using them, and we'll come on to that later. They can produce all of the tissues that are present in us. Once we move beyond a certain developmental stage, and that's very quickly, that's after just a few weeks, we find that the cells that are present in tissues are multipotent, and that's that key phase of saying that they're restricted to being able to make the specialized cells relevant to where they live. Unipotent is an interesting term. you often hear these ones described as progenitors. So when you have a stem cell that's only really capable of making one specialized daughter cell, the oligodendrocyte progenitor cell would be a good example that Robin was alluding to earlier. We often call them progenitor cells. You can think of them as being a stem cell that's unipotent. It can regenerate itself, and it can make one type of specialized cell. So those are the kind of terms that you will come across when you're reading about this. Potency depends on source. My picture's not as sexy as Robin's, I think. I quite liked his picture of getting older. Mine is a bit more, it's just this sort of stick man. I feel rather underprepared now. As I said, that very first few days of life is a really exciting and interesting time, and much of stem cell research that's interested in developmental biology, fundamentals about how human beings are created, how tissues become so specialized and so different from each other, has concentrated traditionally on that early phase, the blastocyst. I'll show you a picture in a minute of what we mean and where they come from. Everything from a fetus onward is essentially multipotent. So that's an important thing to recognize. When you're thinking about stem cells, think about which tissue they came from, and also which developmental stage they came from. The third thing to say, and as Robin emphasised in his talk, is that actually ageing has an effect on how much ability we have to grow them, I guess, how efficient the stem cells can divide. And that's an important thing when we're thinking about using them in the lab or as treatments. So perhaps young tissue-specific stem cells are not quite the same as tissue-specific stem cells in later years. So a number of variables that start to muddy the waters when you read about in the papers a stem cell treatment. You've got to bear in mind they're not all the same. Where they come from is important. The tissue they come from is important. And the age of the person or the animal that they come from is also important. So potency depends on source, or at least it used to depend on source. And you've already heard the big breakthrough in the past 10 years was our ability to take tissues like skin, like gut, like kidney cells, and turn them back into pluripotent stem cells. Really exciting breakthrough. My boss up in Edinburgh always says it gives hope for orthopaedic surgeons that it was an orthopaedic surgeon who discovered it. If you're a doctor, we tend to be a bit sniffy, but orthopaedic surgeons, the solution is usually a bigger hammer. Neurologists are a bit more egocentric and think we know the answers. An orthopaedic surgeon from Japan, Shinya Yamanaka, in 2006 published this revolutionary technique which opened up the door to saying perhaps we now can get pluripotent stem cells from any cell. Terrific breakthrough. So that's a new opportunity, and I'll touch on that in a bit more detail later. That said, source is still important for lots of practical reasons, not least of which ethical implications of how we get them, whether you feel it's right that we get them in this way. 10 years ago, where I'm giving this talk, there wasn't induced pluripotent stem cells. If we wanted pluripotency, we had to go to the embryo. Blastocysts are that very early stage of development, and that bundle of cells you see in the picture called the inner cell mass, just there, that was the bundle of cells that was key to really developing all of the lines of embryonic stem cells that we had. We saw a fascinating split between North America, where the development of new lines was not allowed, and Europe, where the development continued throughout the 90s and 2000s, and we saw big migrations of scientists between those continents looking to do research into fundamental stem cell biology. An illustrative, really, of that first question, ethical acceptability. Where did these come from? Largely, these stem cell lines were derived from in vitro fertilization. As you know, when that process occurs, there's an excess of fertilized embryos which are discarded. That would be the routine practice. In certain countries, for research purposes, we were allowed to take cells and then use them to develop embryonic stem cell lines. And there are a number of embryonic stem cell lines available for researchers to work on. That's where they come from. Given that these are stem cells which are, if you think about it, fundamentally programmed to grow like crazy, this is going to end up looking like one of you one day. That's an amazing thought. What they are driven to do is to grow. So one of the problems we've always had is to think about, if we use these in a therapeutic way, beyond just thinking about can we understand human development and what might go wrong, will it ever be safe? Will these cells continue to grow uncontrolled? Linking obviously to the same kind of problems that you see in cancer. Are these just an opportunity to give our patients a cancer? So that's always been a concern with these. The other issue with these has been that they are very demanding. They are just like having a baby. They need a lot of attention regularly. If you've ever worked with them, you need very green fingers. You need to be in the lab all day, all night, feeding them, watering them. When they cry, they need attention. These cells are hard work. It's always been a job for a very few number of research labs to be able to take them on and actually take them through to a stage where we can begin to look at the potential to develop nerve cells, oligodendrocytes, that we could then do research on. So very challenging cells to work with, ethical problems for many people, and those remain about whether it's reasonable to do so. And there's always that ultimate question about, is it safe to take these forward as a treatment? Along came IPS, groundbreaking research. For those of you who keep in touch with the stem cell news or even just broadly are aware of some of the controversies that go on, this has been a fun area to be around. There's been some advances since 2006. As you might expect, things have changed a little, but this was the original approach to how you take a skin cell and you turn it into something magical that can become anything. What was done is to take those stem cells into the dish and then what's called transfect them, introduce some new genetic material to those cells, which then switched back on this pluripotency. They could then be grown up and then potentially differentiated into the specialized cells that you would like. Alarm bells would always ring when you're changing the genetic material of a cell. That's always been a concern. These cells are fundamentally altered from the way they started. So there became clearly that concern about, could they have different behavior if we were ever to take them forward to be used as a treatment? So concerns also about safety. Since that 2006 breakthrough, which won the Nobel Prize for Yamanaka last year, there have been developments in that now we can achieve the same end of pluripotency without having to change the genetic material simply by introducing relevant factors into the cells of the skin, gut, kidneys, etc. Very difficult thing to do, but important concept that it can be done. So we're really taking off in an interesting direction and opening up new doors there. In terms of the most recent controversy, the thing that's been fun to watch has been this recent publication in January this year in Nature about, can you do this by bathing cells in acid? Some of you might have been aware of that. There's been a huge scientific interest in the potential that a very simple process of bathing cells in acid may be able to induce pluripotency. And since then, much debate about whether that work is valid or not. So you can see that this is a very early phase of research for this. Lots of controversy. Can the results be replicated? And if so, can we take these forward in a way that will be safe to ultimately lead to treatments? How about taking nerve or neural stem cells from fetal tissue? Again, ethical acceptability is in question here. Where these come from is from terminated fetuses, where consent is given. They are used for research purposes. They have been very helpful in us developing cell lines which enable us to better understand human development. That's an important topic of research, but there is no getting away from the source of those tissues and very deep-seated and genuine ethical questions about whether that is a reasonable source of tissue for treatment development. There is the same question about, can we control growth? Fetal stem cells, just like very early embryonic stem cells, are programmed to grow rapidly. Can we control that if we ever were to use that as a treatment? And again, very difficult cells to look after in the laboratory, lots of practical and technical challenges. And then finally, how about from the adult neural stem cells? Robin made the point that the oligodendrocyte progenitors are widely distributed and easily accessible. How about more generic stem cells that may be able to produce either neurons or oligodendrocytes? Yes, there is evidence that those are present. When I was at medical school, we didn't know about those. Those are a fairly recent discovery. They are in very low numbers and very inaccessible areas of the brain, so very challenging to get to. For practical reasons, largely, we've found it difficult to get enough of them to start to do the research that you might wish us to do. Those are the kind of cells that I guess you might label as being important for the classic paradigm of what can you do with stem cells. Could we turn them into something that could then be used to transplant into the nervous system, potentially replacing what has been damaged and lost? How about if we move to a different type of approach? What about using cells to try and provoke the endogenous repair, so the repair systems that we all have, that you heard about in Robin's talk, that decline with time? Is there a mechanism whereby cells could provoke endogenous repair mechanisms to do better? Or could it be that by introducing cells that aren't native to the nervous system, that we can use them in a similar way to what the oligodendrocytes do at the moment, and that's to nourish axons? Could we provide protection in that way? These are exciting questions, and the focus in some ways has switched towards these type of mechanisms over recent years. That's the space in which you'll find there is a lot of commercial interest. There's probably a lot of stuff that you will read in the press around UCB, umbilical cord blood, which I'm sure you may have heard of, so a type of fetal bone marrow stem cells, and adult non-neural stem cells such as mesenchymal stem cells, which again Robin mentioned earlier, and which I'll talk about in some detail later in the talk. So that's what they are. And the important message there really is that they are not one entity. The key property is potency. What can you make from them? And that depends largely on where they come from and the age of the organism when you capture them. What can you do with them, I guess, is the heart of today's talk, that I wanted to really try and give you in a concrete way to say, what is the potential of stem cells? What might they do for you as the MS patient community? The first one, and I've deliberately put it here because actually as scientists it's probably the one that's bearing the most fruit and perhaps likely to bear the most fruit in the immediate horizon, is for us to begin to model disease in our lab, to have enough quantity of nerve cells or of oligodendrocytes, or indeed both, that we can then begin to manipulate and understand is a huge resource for us. We can then begin to do what Robin wanted us to do, and that is to say, can we understand how the car engine works? If we understand that, we stand a chance of being able to repair it. The second thing which has been exciting is if we can have an innumerable number of nerve cells or oligodendrocytes, it offers us the potential to test new treatments quickly. So to test new treatments in a high throughput way in the laboratory, rather than using animals as we have in the past, actually use human tissue and establish whether we think there is any meaningful effect that would be worth exploring in patients. And finally, and perhaps the most exciting possibility, is given that we can now take cells from skin, gut, etc., from individual people, we can then develop individual stem cells. We are all different, so it offers the potential to look at personalizing treatment. So not just screening drugs in a generic way to say, do these drugs have an effect on oligodendrocytes, but actually asking really bespoke questions about, do they have an effect on your oligodendrocytes, which is ultimately the big movement in 21st century medicine, to try and personalize things, to say we know the drug works in a group of people, but do we know that it works on this person who's come to see me today? So really exciting prospects for just having a resource that we can do these kind of research projects on. The second thing, and I think perhaps what is often held as the holy grail when one originally began thinking about stem cells, is can we switch off the disease? Can we reset the immune system? And the paradigm for how that's been approached is very similar to what you would see in cancer treatments such as leukemia, etc. It is a bone marrow transplant, given that bone marrow is where the white blood cells are made, after ablation of the existing immune system. So it's a very tough treatment to have. You can imagine that the picture shows you it's quite a dramatic thing to have. So patients have mobilization of their stem cells at the beginning, capture of those stem cells, and then go through very heavy-duty chemotherapy to kill off the existing immune cells. Those are then replaced by transplantation of hematological stem cells, and the hope would be that when the immune system reconstitutes with that transplantation, we don't have the same autoreactive population of cells. Now, there is a literature on that. There have been lots of relatively small phase 2 studies done over the past decade or so, About 800 patients with MS have been treated with this, and the results are interesting. Certainly it's very effective in stopping relapse activity. That's been exciting. Whether that's aggressive chemotherapy or the cells, unclear. What's been less exciting and perhaps a little disappointing has been following those patients up over the longer term and seeing that disability continue to progress. So we have a very toxic treatment and an uncertain benefit at the moment, but I think there is possibly more research to do, excuse me, and we shall know more in time. Excuse me, could I have a glass of water? Thank you. That's better, I hope. The third mechanism for how these cells may be useful is to be used as a Trojan horse to deliver local therapies to areas of the brain where we can target them. Now, this picture here is of the Kelpies. It is a beautiful sculpture in Falkirk in Scotland, just near where I do clinics on a Friday. I drive past it and it always reminds me of this mechanism of how cells might be used. This is very much in the earliest stages of development, but the idea here is very interesting. So the idea with this approach is to say that what we have on our hands with stem cells is a mini factory, that we could ask these cells or instruct them to make or deliver local therapies and allowing them to target the areas of the brain which are inflamed. Many of them already have that as an existing property, to be able to migrate to areas where inflammation is active. Therefore, it's a fascinating potential technique to get drugs at high concentration to the parts of the brain where they may be relevant. Very early, but it's certainly a potential route for using cell therapies. The fourth mechanism of what they might be useful is, I think, probably the one that everyone thinks of, and that's to repair the nervous system. And it's worth splitting that into two types, which have already been touched on by Robin in his previous talk. Let's be really explicit about that so that we all go home being clear and with a clear idea of what we mean. The first approach to repairing the nervous system is obviously to say that we have lost tissue, let us replace tissue with that which we've grown outside the body in a lab. That would be called exogenous repair, and that's the classic example of what we all think of. Can we take new oligodendrocytes, put them into the damaged area, and allow them to do what we would hope them to do? That's clearly the most challenging and perhaps the most distant objective of all stem cell research. Why is it the most challenging and distant? Well, the brain is the most complex object in the known universe, and MS happens to be the most complex disease of the most complex object in the known universe. And so that makes it tremendously interesting to work in MS. It also makes it tremendously difficult to find solutions to that. We have fundamental problems in getting cells to the right place because the damage is everywhere, at lots of different sites, at lots of different times. We have fundamental difficulties of instructing the cells to change into the right kind of cells once they get to where we want them to be. And we also have a fundamental challenge to control that once they're there. Are they going to be functionally active? So even if we could turn them into an oligodendrocyte in the place in your nervous system where we would want them to be, are they going to function when they're there? And are they going to function normally? So huge challenges to actually implement this type of research. But it's always been the holy grail, if you like, of stem cell research. And I think it's still the paradigm that everyone thinks of. It just happens to be the most challenging and the furthest away from being delivered. So how about as an alternative? Could cells actually orchestrate what's going on in the repair pathways? So if you envisage here, you've got this beautiful orchestra of repair systems within the brain, which works very well most of the time. However, as we know and as we heard about earlier, it declines in its function over time and eventually it stops working. Could we bring in new cells to try and orchestrate those repair systems better, to try and get them functioning as they once did when we were all young, fit, handsome, etc.? That's become the real space where a lot of stem cell therapeutic research has moved into. Are cells a clever way of doing that? Or are they a way of at least illustrating the concept that it can be done, which would then allow us to go back and think about what was it that they did? What were the steps that they triggered that allowed the repair system to kick back in? And finally, could we use them as a way to protect the nervous system from ongoing disease, either by reducing disease activity, just like your DMTs do? Could it be that they're just as effective as your beta interferon or alemtuzumab or natalizumab? Might they be a good option for that? Could we use them to restore myelin integrity, remembering that if we do that, that is in of itself protective to the axons? And could we use them as a substitute for oligodendrocytes to give out those trophic factors, those nutrient factors that nourish the axons when they're in a vulnerable state of having lost their myelin? So these are the 5 main areas. I'll put them back up for us because I think that's crucial that we take home as to what can cells do. Let's run through them again and think about really how active is the research in these areas and where's it going? Well, what's happening in these topics at the moment? So as you probably picked up from me before my voice died, modelling disease in a dish is really flourishing. This new wave of induced pluripotent stem cell technologies has really engaged a huge amount of research activity in that. It's early research activity, but it's hugely promising in those 3 areas that I mentioned, and they're all being explored. So we are beginning to unpick, by now having in a dish oligodendrocytes for the first time in a reliable way, what can go wrong with them. We are now able to start to screen our drugs in that setting. We are now able to look at individual patients and explore whether there are differences in your oligodendrocytes. That's a very interesting idea. We're all genetically different. Is it even safe to assume that an oligodendrocyte from me is the same as an oligodendrocyte from you? Perhaps not. These questions can begin to be addressed now, and that's a hugely promising area for research, finding out how the engine works and what goes wrong with it so that we can then think about fixing it. Resetting the immune system, I think there's more to say on this. I suspect there is a story still to tell, even though there is 15 years of effort has gone into it. Very toxic treatments at the moment. Benefit on relapses but not progression is an interesting thing. I suspect we will learn more about how the disease operates from this kind of research. But actually, as you heard yesterday, we have very good DMTs now. Can I see a role for them competing with some of those? It may be challenging for them to really oust some of the big boys in the DMT arena. As a Trojan horse for drug delivery, really interesting, very much in infancy. No one is really going into patients with this approach yet, but there's a lot of preclinical work beginning to happen about how that might be utilised, how we might make more oomph from the drugs that we've already got, and how we can get them to the right place. Repairing the damaged central nervous system active, I'll tell you about that in a minute, and protecting the CNS from the effects of disease also active. And these 2 latter ones are what I wanted to give you some information about in terms of a small stem cell therapy trial that I've been involved in. So let me tell you a bit about that now. So reminding you that when someone says I was involved in a stem cell trial, what you should say to them is, okay, what do you mean stem cell? So let me try and answer that question, as I'm sure you are all just about to answer me. What I mean in this context was that we did a very small proof of concept study in Cambridge, which I was involved with, looking at mesenchymal stem cells, which are a population of stem cells in bone marrow and which are of enormous interest because there's been commercial interest in that, and I'm sure you're aware of that. There's been a lot of preclinical work on whether it could be helpful in MS. So we set up a small study in secondary progressive MS of 10 patients with a single treatment. I won't tell you too much about the pre-trial, post-trial design. It brings out my inner geek and I waffle on for a long time. What happened was we followed patients for a period of time to watch their MS. We then gave them a treatment and followed them up for a period of time, and guess what? We watched their MS. What we were looking to do was compare before treatment to after treatment. We were particularly interested in this trial of looking at the visual pathway, really just because it's very difficult to measure MS in patients. Focusing on one area is quite a useful way to try and get a handle on what has happened to my patient. We aimed really just to prove that it can be done and that it was safe. All of the caveats that you've heard from me about stem cells have included safety. That's always been a concern, and that was our primary concern, as always in these trials. Is it safe to give people a dose of stem cells from their bone marrow? What will happen to them? Clearly, the proof of concept is, does it have any effect on the brain tissue itself? And you can see from the picture there, what I was hoping to show you and what we were exploring in the efficacy is whether when we follow patients before treatment, we can watch them getting worse, declining over time, and whether after treatment we can either level that off or even improve it. We looked at around 100 patients to capture our 10 who were eligible for it, so it was a difficult trial to find the right patients. We were very fussy, and there was a huge amount of interest in that, as you might imagine. But we got to our 10, and they stuck with us the whole way, which actually is a huge credit to the commitment, I think, of patients with MS. This was a very difficult trial to participate in. They had bone marrow samples taken, they had huge amounts of imaging assessment. It was a challenging trial for people who were quite disabled, so I'm hugely in their debt and take my hat off to the resolution of these people to stick with us. We had 10, we had 7 boys, 3 girls, around 50 years of age, typical secondary progressive group of patients that we would see in our clinic, really. They had had MS for around 15 years. The EDSS, for those of you who know that scale, the average patient would walk with 1 stick, and that was in the upper range of where we were targeting. They all had damage to their visual pathways. That was what we were fundamentally interested in. But it wasn't new. It wasn't that they had just had an optic neuritis. It was some time ago, because what we were really interested in isn't going in the fresh situation to try and improve repair, which was going to happen anyway, but actually when things had settled down, when the dust had settled, is there anything that we could do to change matters? So this is a fun slide, which is hopefully meaningless. If it means anything to anyone, then come and see me later. You can tell me more. We spent a huge amount of time characterizing these cells, and I really put it up purely to show you that this is a tricky thing to do. We had the Professor of Pathology helping us out with some of this in Cambridge. And what it shows is that we went to some length to prove our cells were what we said they were. I've told you that stem cells can make daughter cells which are specific to the tissue they're in. So what you're looking at is the red blobs are fat cells. We've turned our stem cells into fat. The brown stuff is bone in the middle, panel B. We've turned our stem cells into bone. And the blue stuff is cartilage. We've turned our stem cells into cartilage, and that was all we could turn them into. And that fits with what we would expect for these cells. The wiggly line at the bottom is us looking at the genetic material of these cells. What you must remember is that I spent 8 weeks growing them in a laboratory, so they were expanded about 10 billion fold. So the potential for mutations and for us to introduce cells which were not quite what we took from the patient was clearly there. If you introduce mutated cells, there's clearly a concern about them being abnormal and potential for malignancy, cancer, etc. We had to show that they were normal in terms of the DNA, and we did that too. Hard work. What did we find? Well, I'm pleased to say our primary objective was great. They were very safe. We managed to culture these cells in all of the patients we tried, so it can be done. That's the first thing to say. I can take your cells and grow them to a pure population of what we want. When we gave them, what we saw was typical of a mild hypersensitivity, or if you like, allergic reaction. One of our patients got a rash around about 12 hours after the infusion. It got better after about 24 hours, didn't require any treatment. Another patient had an itchy scalp a week later, very difficult to know whether it's related or not, again settled down within a week without treatment, so very safe, at least in this small group of patients. More importantly, given we had expanded these cells over time, did we give them any nasty infections? Did we give them any evidence of abnormal cell growth? No, we didn't, and that was then followed up for around 10 months afterwards. Follow-up continues now, and again, no long-term adverse events have been reported. We're now up around about 5 years now after that trial closed. The bit you're interested in is what happened to their eyes. So vision chart, as your optician goes for, is on the left there, and you can see that the higher up the chart you go, the worse your vision. What we found before treatment is this dotted line here, was that our patients were very slowly getting worse. So on average, our patient could see only further up the chart as time went by. In other words, the vision was deteriorating very slowly. After treatment, we saw quite a sharp downturn. Their vision improved. That was exciting. But clearly that was only clinical findings. All of our patients had treatment. Perhaps we were biased and just pushing them to try a little harder. That's always the concern you have when you don't have a blinded trial and you don't have a control group. So how about blinded assessments? Looking at the function of the nerves with visually evoked responses, the physiology of how quickly they could conduct and how much signal could conduct through the visual pathways. We found that after treatment, the speed of conduction got a lot faster in the visual pathways. We found that the amount of signal getting through to the back of the brain at the visual cortex had increased too, so really encouraging. And when we looked at the actual structure of the optic nerves, structure of the visual pathways, we found what you're looking at is the optic nerve, this circle here. I always think this looks a bit like the Terminator, those of you who are old enough to have watched the Terminator. It scares my children. I can't show this talk to them. What we found was that before treatment, the optic nerves were shrinking, as you would expect, nerves shrinking over time. And we found that that was reversed after treatment and that the size of the nerves was enlarged. So really exciting evidence of clinical improvement, physiological improvement, and structural improvement at the time of treatment, all in a direction which we would hope in the visual pathways. Exciting stuff. How about their global disability status? And again, that score that we use, the EDSS, is a very rough and ready. It's not terribly sensitive, but for what it's worth, we found that patients over time were becoming more disabled before we treated them, and then after treatment, that all leveled off. The brain structure, we looked also at lesions within the brain. What we found, I think, is really fascinating, and I think this is where we can get into a slightly more speculative space. In the brain imaging, you'll know that we can see the areas of inflammation, T2 lesions, on a brain scan. We didn't make any difference to that. Patients continued to get new inflammatory lesions. What we did make a difference to was what's called black holes or T1 lesions, which are areas of tissue loss. They shrunk after treatment. So if we're thinking about what did we actually do, these cells clearly had some effect. What was their effect? Then the preclinical work opened up three possibilities. Perhaps we had promoted being like the orchestra, conducting it, encouraging endogenous repair in the visual pathways. Perhaps we had just affected the immune system, and so we allowed the space for the nerve system to repair. Or perhaps what we'd done is we'd put cells in the right place to protect the nerve cells. So, don't think it was immunomodulation. We didn't have any effect on disease activity in terms of lesions, and we saw an increase in the optic nerve area. An increase in the area usually suggests more inflammation, if that's what it is. So I don't think we had much of an effect on that. In other words, it wasn't like giving them beta interferon. That didn't happen. How about local trophic support? Well, unlikely to be that, given that we reduced the visually evoked response latency, suggesting that actually the myelin was functioning better, not just the axons, the myelin was functioning better. We also saw a change in a type of MRI sequence which suggested there was more myelin there. So unlikely to be just trophic support. So what we're left with by elimination was perhaps what we had done is that we had been like the conductor of the orchestra, promoting endogenous repair to kick in and work better than it had in the past. Very exciting. What I'm saying there is that perhaps what we had done is we had taken these naked axons and somehow, instead of them then degenerating, swapped them back to being in a position where they had their myelin sheaths again. So what I've showed you so far was a change in a straight line which looked as if it would continue forever. So if you follow that out logically, what you would expect is that come the Commonwealth Games in 2014, my patients should all be winning the gold. Clearly that's unlikely. And indeed, most of the patients in the trial were really switched on and asked me repeatedly, how long is this going to last? If it makes any difference, how long will it last for? Will I need another treatment at some point in the future? Kind of obvious question, but really insightful question. The answer, as best we can tell, is probably the effect we saw lasted around 6 months. what we see when we look at different ways of modelling the results that we had is we do see that the best fit is this curve, at which the top of the curve is around about 5.5, 6 months. After that, it looks like we hadn't switched the disease off in a permanent way. The disease came back. So food for thought. Great steps forward, but perhaps not a permanent solution. So I'll finish on this slide. This is my last slide. I'm sorry to have gone on over time. And this is really what next for stem cell research in MS. And again, just going back to those 5 big themes of how they may be effective, I think these are the themes that we will explore over time. The question is really where we take it, and I guess that depends a bit on what you guys want us to what direction you want us to take it in. Modelling disease in the dish has got a tremendous momentum now, and I think you will see results from that over the next 10 to 20 years. Even before that, you'll hear about big splashes which will need replication. That's really exciting, and being in the field, that's where I suspect you'll hear the first news coming from. Resetting the immune system, you can still join trials which are looking at that. They're still going on. My personal view is that we have very good treatments now to manage the immunological aspects of MS, and I suspect that cell therapies may not be the dominant ones that solve that conundrum. As a Trojan horse for drug delivery, I think is on the horizon. That's the one that's probably most distant but really exciting. We shall see. Repairing the damaged central nervous system and protecting it are these 2 ones which I guess are the ones which we all sort of think about and we're excited by. That's the ones which there is some proof of concept now. I'm partly guilty for doing that, but others are engaging with that in a big way now. So I think that space is active. And over to you, really, to hear about what the kind of things you would like us to do with it, where you'd like us to go, and what sorts of trials you would like us to do. Thank you very much for listening.
Peter, thank you very much. It's another fascinating talk. We do have time for questions if there are hands going up. Jenna, there's one right behind you, right at the back.
Well, yes, very pioneering work, but I just want to question the robustness of the data you presented, because it was a very small group, only 10 individuals, it was not blinded, and also there was no control group. So are there any other explanations for the data that you presented other than the therapies that you used?
Yes, it's a great point. So you're going to unlock my inner geek about the methodology now. I must restrain myself, but you're absolutely right. The kind of trial we did is got this technical term of a pre-test, post-test design. So what we had was the patients were historical controls for themselves. So we had a historical period before we treated them, and then a period after treatment. Now, at a very fundamental level, you can't ever definitively prove that any change you see was because of what you did at that point in time, because other things could have changed too. So that's a fundamental point. So I think all we can say is that this early pilot study really is suggestive that there's something of interest in following up with more robust and larger controlled and blinded trials. However, you're quite right that I wouldn't wish to oversell it and say we have any definitive evidence. What we found is certainly consistent with a change at the time of treatment, and a very exciting change, but clearly there's more work to do to prove that in a definitive way. On the point of blinding, there was blinding for the physiological measures and the imaging measures, in that those assessments, so when we were looking at the scans and measuring how much inflammation there was there, we didn't know whether this scan was from before treatment or after treatment. That aspect was blinded, so we can be a bit more confident that changes seen in those aspects of the outcomes were genuinely occurring at the time of treatment. The more fundamental point is, can you attribute the change to the treatment, not in this type of trial, which opens up an exciting opportunity to do it in a slightly different way with a larger group of patients.
More research needed. I can't currently see a hand, but I wanted... Oh, no, actually there's several. There's one at the back and then one towards... two at the front. OK, we'll start at the back.
I've been reading a lot about a lot of Australian patients with MS who are actually paying a lot of money to go to Russia to have their, like a chemo session, and then their immune system's reset. Mm-hmm. Does that happen in this country at all?
Sorry, I can't quite see you. Hello. Yes. Only in the context of research in this country. So there's international research efforts looking at hematological stem cell transplantation for that second example that I gave of how stem cells might work, which is to try and ablate the immune system that we all have, which we believe is targeting the nervous system and causing the damage, and replace it with an immune system which one hopes will not do the same, one hopes will stop targeting. It's a research tool in Europe, out with the regulatory environment of the European Medical Agency and the equivalents elsewhere, such as the FDA. There are pockets of commercial activity around the world. I think you mentioned Russia, that's certainly one of them. There are others too. Personally, what I find is I often get individual requests from patients emailed through to me or sent through to me asking what I think about these things, and my response is often the same, which is to say that there isn't the evidence yet to say that that is a helpful thing to do. We don't know enough about what is being done often with these commercial treatments to say that it's even a safe thing to do. So my advice is often not to explore those options and to look for if there is any research available to you that you might be able to participate in if it's something that you feel strongly is of interest.
And this is an area where there are active clinical trials, so using the clinicaltrials.gov website is always a good route through, rather than going off to clinics that don't necessarily follow the same safety protocols as they would in the UK. We had one at the front.
I didn't get the whole thing, sorry, I arrived late. If you make an infusion and you see an improvement, then it stalls to a plateau. If you were to do another infusion at that point, could you grow an S-shaped curve such that at some point you would end up with a repaired tissue rather than partially?
Great question, and it's unknown. So what we showed with that slide, which was trying to answer the question that, as you've just put it, all my patients were asking me in the trial, is this a permanent change or is it temporary? It's temporary. To discover whether you can then get further improvements and when it eventually plateaus and whether there is a long-term fix, essentially that would take a study where repeat treatments were given. It's an interesting question. It hasn't been answered yet.
What if you were to do infusions over a regular basis with the patients that you've got, because obviously you've got stem cells from those people, one assumes that you've got to keep them cryogenically of some point to hold them. And if you were to infuse them again to see if that next step happened and what level of the next step, because I wouldn't expect it to be a true S-curve, I'd expect it to slow down. So it might be a shallower S, but what point can you reach where it improves the quality of life of the person such that, you know, could you make a blind man see? Is it a case of you've just made the chemical conductivity of the optic nerve better? Have you improved the actual nerve itself? Or have you improved just the protection and everything else is still tickety-boo doing whatever it is? Or have you reset everything along the way?
So there's quite a lot to unpack there.
Sorry.
It's a great question.
If I could answer any one of those, it'd be a Nobel Prize.
Well, that's it, isn't it? So to begin with the fundamental question of what would happen if we did it again and again and again, no one knows. That would require doing it and finding out. But it is certainly a hypothesis that could be tested, that you would find that you may get plateauing or eventual failure of treatment, or indeed you might see an improvement to a level where it's stable and you enjoy good function. We simply don't know. That's not been tried before. And actually, when it's used in animal models, that kind of approach of giving two treatments, you get that S-shaped curve that you see, but in humans it's never been tested. So interesting. The second question is about the mechanism of if these did indeed have an effect, and this wasn't a spurious finding, as I would contest that it probably wasn't. How did they have an effect? What is the mechanism? I think is fascinating. Ultimately, in this study, we can't definitively answer that. What I can say to that is what the data suggests. So the data suggests that we did not switch off inflammation. So the brains of the patients continued to acquire inflammatory lesions at the same rate as they did before treatment. So we didn't do that. We didn't appear to just have a trophic nourishing effect on the axons, because what we saw the nerve cells, certainly in the anterior visual pathways, enlarge, and we saw the black holes, the areas of tissue loss within the brain, shrink. So it looks like there was an effect on structure within the nervous system. And thirdly, what was the cell that we influenced? That's the hardest one to answer. But if one looks at the physiology, we see that the conduction speeded up as well as got to a greater volume, if you like, at the back of the brain, suggesting that we weren't the tortoise, we were the cheetah, which suggests that perhaps the myelin was functioning better. There's lots of ifs, lots of buts, but that's where the data takes us in terms of if we can speculate on what the mechanism of effect that we saw, it's suggestive that perhaps we were orchestrating the repair systems that were within the brain to do a little better than they had been doing previously.
Good. I'd like to move on to another question, if that's all right. Kim, there was a question right in the middle, who I believe is the chair of our Sutton branch.
Yes, thank you, Chair, like I say, of Sutton branch, also a lapsed immunologist or immunotoxicologist for a while. So hence I fully appreciate that research is a long-term thing. If you were to stand here again in 10 years' time, what would be your hope or your top-line message in 10 years?
So I think we've had fundamentally we've asked the right questions from the floor already as to what the questions that I'm keen to answer is, first of all, as was asked at the back, is this effect real? I think that's the first thing to say. That would require a study showing in a blinded, randomized trial that we do see the same changes. So that would be the first thing. Is this a real effect that we can see? Second, I'm curious to know the mechanism of effect. Is that we have promoted endogenous repair? And third, I guess, is to kind of unpack that and say, well, what exactly is it that the cells have done? And what's called reverse translation then, which is to then take the MSCs back to our disease models and look and see what were the altered biology that we've achieved by introducing these that resulted in us orchestrating the repair system, if indeed that's what's happened. So I think that tiered approach is probably how I would see this research going. We always want to run before we can walk, so clearly there's an impetus, there's a natural impetus now to think about a bigger trial, moving on to phase 2B, thinking about even phase 3 down the line, planning for it at least conceptually. I think there's some work to do exactly around these questions of, is it real in a definitive phase 2? If it's real in a definitive phase 2, how long does it last for? And can we really nail the mechanism? I think those are key before we rush forward. But it's always nice to give some grounding to say that actually that is a strand of research which looks certainly worthy of exploring, if nothing more than that.
Peter, this is an area where there's tremendous public interest in stem cell therapy, and so it gets quite a lot of media coverage. Does that make it easier for a stem cell scientist, or does it make your life harder?
Well, we live with it. As you know, there are only two outcomes in the popular media for any stem cell trial, which is disaster or miracle. I'd like to argue that mine was neither, really. There was certainly no disaster. There wasn't any miracle cure, but what there was proof of concept that we changed something. Questions about how and for how long remain. So we often find ourselves doing that very scientific thing of saying, actually, a more measured interpretation is what's required. So when we engage with the media, we're always coming across as complete nihilists. I'm sorry about that. We're not really nihilists. We're just trying to be respectful to the data that we find and actually try to answer the questions that it generates as much as anything else, and not give false hope where that's inappropriate.
Good, thank you. So I'm now going to get to the point where I have to thank you for coming. But as I'd said earlier, thank you also for all the work that I know many of you do across the country, being activists in a sense for the MS Society and helping us to raise the money that enables Peter and others to do their work. Thank you for all of that. A reminder that there are places outside. Somebody came up to me earlier after a session and said that actually they find some of these research talks both something that they're really positive about, but there's always things that they really want to talk to someone about. So there are people outside who can talk to you, both the scientists, but also others who can talk to you from a more emotional perspective if that's useful to you. But also for now, the main thing for me to thank Peter for coming, for giving up your time today, and for the work you do every day on all of our behalf. Thank you.
Thank you.
Well, welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLife2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Live that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. I'm watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Live experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Live, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Life experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Life, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening, you'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on.
Watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. I'm watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. I'm watching everything that life gives to me. I stand tall.
Go.
Hi, I'm Stuart from the digital team at the MS Society. This weekend, along with the rest of the team, we'll be at MS Live 2014 in Manchester. We'll be bringing the MS Live experience to you on Facebook, Twitter and Instagram. We'll be interviewing speakers, reporting on events and capturing all the best bits. But we can't be everywhere at once, so we need your help to decide where we should go. How? Check the schedule, then use the hashtag #SendStew to direct me via Twitter and our Facebook to anywhere at MS Live 2014. For example, send Stu to the wheelchair dancing in the Get Active zone, or send Stu to watch the ice cold chef cooking demo in the MS Live kitchen. Obviously, we'll try to meet your requests within reason. If you say send Stu to Cornwall, Coventry or Guadalajara, we may not have the time or budget to do it. If you're coming to MS Live, get involved by letting us know what you've been up to and sharing your thoughts and photos by using the hashtag MSLive2014. Most importantly, we want you to be ready to join in, so follow us on Twitter, Facebook, Instagram, Google+, and YouTube. See you at the weekend. And sharing your thoughts and photos by using the hashtag MSLife2014. What? Welcome to MS Live 2014 in Manchester. So here we are. Tomorrow, when all the action will be happening. You'll be able to watch the research talks. We'll be live streaming them, so you'll be able to watch them as they happen. Go to the website for more details to see what the talks are about and what time they're on. You'll be able to watch them live. And yes, if you have any questions, go to the website. And if you want to see something here, but you can't be here, I'm Stuart. You use the hashtag #SendStew, and you can send me to see a bit of MS Life that you can't see. And I'll maybe go there. Thank you. I'm going to ask him how he feels about MS Live. Craig, are you excited about MS Live? Yeah, he is. So, yeah, moving on. I stand tall. I stand tall. Head held high.
I stand tall, watching everything that life gives to me. I stand tall, my feet are rooted to the earth like a strong oak tree. I stand tall, the wind blows. I stand tall, I stand tall, my life grows. I stand tall, I stand tall. I'm watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall. I stand tall. I'm watching everything that life gives to me. I stand tall. My feet are rooted to the earth like a strong oak tree. I stand tall. The wind blows. I stand tall. I stand tall. My life grows. I stand tall.
Welcome to MS Live 2014. I'm with Shaa Pizaro, who is going to be doing a talk, Escaping Labels. Hello, Shaa.
Hello.
Could you tell us a little bit about the talk and what you're going to be talking to people about?
Well, I'm going to be talking about a postcard that I entered into a project called Postcards from the Edges. And the brief was, what would you want to say to the world about being disabled? Put it on a postcard. And I sort of laughed, and there was a picture of me smoking in a back alley, and I wrote, I drink, I smoke, I swear, on it, sorry, and sent it off. And it got a lot of press attention, and it's been, you know, travelling around the country in an exhibition and ended up in Parliament. Fortunately, I know a lot of politicians quite well, and just said, oh, it's what you'd expect from me, and they laughed. So, yeah, so I'm talking about that, and I'm talking about why I did that postcard, and actually what's behind it in terms of dealing with a diagnosis with MS, coming to terms with being disabled, and, you know, the way that MS affects, you know, my life, and the way that I've kind of overcome that to make sure that I still get to be me.
Now, the talk you're doing is Escaping Labels. Could you talk to me a little bit about what labels you're thinking of escaping?
Well, I talk a bit about how disabled people are represented in the media, because I think that very much there's sort of three categories that the media present disabled people in, and one is Paralympian supergods. The second is liars and scroungers and benefit cheats, and the third is sort of tragic cases that need to be pitted. And I don't think I'm any of them. And I don't think most disabled people are any of those. And so, yeah, I want to talk a bit about that.
Also in the talk, it's talking about low self-confidence. Could you give some tips about low self-confidence and how to overcome that?
Yeah, I was in a marriage to someone who couldn't cope at all with the diagnosis of MS. And he very vocally would tell me that I was disgusting because I was disabled and that he couldn't find me sexually attractive, knowing that I had to catheterize. He was really very abusive. And in fact, my divorce got put through as a priority case on the grounds of abuse. I knew that I couldn't handle that myself, and I couldn't see how I could ever sort of regain my self-esteem and my self-confidence without getting professional help. And so I went off to a counsellor, and I found the counsellor that was right for me, and I spent 6 months with him. And at the start of it, I said, I want to believe that I'm still me, and that I am sexy, and that I can, still be found attractive. And through seeking that professional help, that was the thing that really changed things for me. So I think that would be my real advice to people, is seek out and accept the help that you're offered.
Thank you very much. And I think everyone could agree that Shana's looking very sexy today. Welcome to MS Life 2014. We're about to speak to Trevis Gleon. Trevis, you've just done a cooking demonstration today. Could you tell us what you cooked?
Cooking is something that can take up so much energy, and eating is something that we really do need to do 2 or 3 or 4 times a day. Putting MS into that mix can be very difficult. So what I did today was what I call cooking without cooking. So it's for those times when you're absolutely knackered and you just don't have the energy, and beans on toast or a poached egg and toast, again, isn't really what you want. So I showed everyone how to make a whole poached chicken, where all you actually have to do is put the chicken into a pot of water, bring it to a boil, put the lid on and pull it off the hob, turn the heat off, and then go have a nap. An hour and a half or 2 hours later, the chicken is completely cooked. I worked in the industry for probably about 15 years before MS sort of ripped that away from me. Learning to adapt my love for food into my abilities, energy level and physical abilities and all that has been part of what I call getting my heart around living with MS. I always say that it took me about 5 years to get my head around the disease, multiple sclerosis, and only then could I get my heart around actually living with MS.
So what tips would you give to people about cooking when you have MS?
On the days that you have a decent amount of energy, or that there are an extra pair of hands or two in the house, maybe you peel all the carrots for the week. Maybe you peel a few onions in advance when you have some extra energy. Things like that. You can purchase now good quality products that some of that work has been done. The washing has been done, the dicing has been done. There are good quality frozen vegetables now. I mean, I grew up, most of the vegetables we ate were out of tins, and it was almost a joy to get a frozen vegetable. Then I became a chef and, turned my nose up on these things. But they can be very good quality products that are picked and processed at the height of ripeness. Adapting your menu to the level of cooking that you can do. And the dish that we did, the savory porridge, is a perfect example of whatever is in the house that can go into that dish, goes into that dish. So it's a matter of stocking a really healthy larder so that you have ingredients you can choose from, having ingredients either in the fridge or in the freezer that you can pull out and use whenever. And then also, it's not a bad thing to cook ahead and put a couple of portions in the freezer for the days that, the microwave is all you have.
I think we have some very practical tips there. Very useful to me too.
You don't have to have MS.
sometimes you don't always understand him, but I thought he was really good. Because sometimes you get a neurologist and they're like a little bit uppy on you.
Yes, yes.
And you're like, oh God.
Yeah, that's what I had when I was diagnosed.
Yeah.
When I was diagnosed, that's what I had. Well, the neurologist we've got now is younger and they've got a different, completely different manner, much more approach when talk about. Yes, yes.
Yeah, because I remember when I was in my training, the neurologist put fear of God into you.
Yeah.
Now I tell them off.
Yeah.
Whereas 30 years ago, they'd been like, no, I'd have I'd gone, oh, yes, slow down and slow down. Oh, shit, we're doing it. Oh, we're not on.
Good afternoon. My name is Rona McLean. I'm the Programme Lead for Education with the MS Society. I'm delighted to be chairing this session this afternoon on fatigue research. Most people with MS will experience fatigue at some point. It can be one of the most difficult symptoms to deal with as it's invisible and can really affect the quality of life. Research into developing fatigue managing strategies for people with MS has increased, and we're now seeing some positive results. In this session, you're going to hear from Adrienne Cox, an MS nurse, about the ways that she helps people manage their fatigue. Then, Dr. Sarah Thomas will talk about her research into developing a fatigue management programme and rolling it out so people with MS can access it. Finally, Tim Warner will talk to us about his experience of fatigue management and how he uses strategies to help him stay active. Before I ask Adrienne to stand up and talk to you, I've known Adrienne for years. She's been an MS nurse for a lot of years, and I used to work alongside her. She is currently working at Facets Community Trust, and she runs a lot of fatigue management programmes, both as groups and one-to-one. So I'll hand over to Adrienne now. Thank you.
Good afternoon, everybody. Obviously, if you've been in my session just before, you'll already have been introduced to me, but I work in Basset Law, which is a very small part of Nottinghamshire, and its main towns are Retford and Worksop. It's very rural in parts and reaches up to Lincolnshire, and it borders onto South Yorkshire with Doncaster and Sheffield and Rotherham, so it sits quite in the middle. Not surprisingly, fatigue is one of the main symptoms that I find, and it affects everyone individually. Everyone copes with it in a different way, but the majority try at first to beat it without much success. I offer fatigue programmes, and before I had Facets training, which is going to be talked about more, I used to use my own programme. I offer group sessions, and problems with group sessions are, unfortunately, in this day and age of the NHS, we do have to pay for a room out in the community, unless we're very lucky and have got our own room in our PCT. And also, group sessions, you have to take into account how far patients have to travel. And being a rural area, it relies on their own transport, because some buses in certain areas are only every hour or every 2 hours. I also offer a one-to-one sessions, because predominantly my role is home visits, so I don't do very many clinics out there. But the problems with one-to-ones are we're not able to share hints and tips with a group. And tips with others is sometimes more important than actually what the professional can give you. What I thought I'd do is I'd actually highlight a couple of my patients and their attitudes to fatigue and what happened when we sort of helping them. So my first one is about Debbie. She had to stop working as a teacher because of her fatigue, and she described it as corridor syndrome, where when she walked out of her room at school, she found a very, very wide corridor and she had to make it to the staff room, and she found this really, really difficult. She also said that her children in her class had the best spellings tests ever because she lost track of what she was doing, and she used to say, right, we're going to have a spellings test, because it was a lot easier for her to manage than her programme, and she realised she needed to stop work due to her fatigue. However, she stopped working, and so decided to fill her time because she suddenly became quite well. Her fatigue improved because she wasn't working, so she started to feel guilty. So from being a full-time teacher, she then started doing craft work. She started painting, as in painting pictures. She learnt to play the piano. She started looking after her grandchildren. So not surprisingly, her fatigue came back and she had to be reminded why she'd stopped work. Because she looked well and hid it from her family, the family did not know how she was feeling. And unfortunately, she ended up having a really bad relapse of her MS and has now had to relook at the fatigue programme all again and about pacing herself and still being able to enjoy her activities. We've started doing a Facets programme with her, and she's already had a really good day out with her husband because she paced herself. Another one of mine was a lady called Julia, who is extremely house proud to the extent where she cleans up before I arrive. I don't know why, because her house is beautiful. She always wants to have everything done in one day and would then find that she was not well for several days. She recently took on board what we'd said about fatigue management and actually paced herself, and I'm very pleased to say that she actually decorated a room and actually allowed it to last a whole week with the mess of decorating. So she's actually taken it on board. Nearly killed her, but she's managed to do it. Another one, Sally Ann, she's had MS since she was in her early 20s and never accepted it. And now she's in her 50s, she realised how much she needed to look at her lifestyle and plan and pace and enjoy. And I only visited her very recently, and she showed me her new kitchen. And actually was very, very proud that it had actually taken her 6 weeks to actually finish and complete it. She's had some help, obviously, but she did play a part in it because she planned. And what she got was the cupboards that she put up. Each individual portion of the cupboard was put on a table in her kitchen, and she was able to paint it. And she said it didn't matter if it took her at 8 o'clock at night when she was feeling a lot better to actually paint one door or one frame of her kitchen. And that has taken a lot of effort for her to be able to do that. But again, she's also now paying for a gardener, so as to mow the lawn, because that was a big issue with the fatigue. And now she says she can just do her flower pots, which she really enjoys. But again, the pacing and the planning of something that has actually had to be brought into her fatigue management. On the other side, I had somebody who was actually made redundant, so she was at home a lot more. She did quite a lot of tidying up. She looked after quite a few animals. She's got chickens and things like that. And she found her fatigue quite bad. But then her husband was actually made redundant as well. But he took over everything. He took over the housework and the cooking. And her fatigue actually got a lot worse. And they couldn't quite understand this because they thought they'd planned. But because she was not doing anything, she was sleeping too much, she was staying up late, and her whole body clock became confused. So the fatigue programme helped her to see that she was bored, she was sleeping too much, and had absolutely no plan for the day. Now her and her husband are working in partnership, and her fatigue is actually improving. So fatigue management can work, but it's very much a learning to be self-management and being prepared for relapses in the fatigue and going back over all the time what you have learnt. Thank you.
We'll be taking questions at the end for all the speakers. I should have mentioned that at the beginning. Thank you. The next part of this session is going to be Sarah Thomas. Sarah Thomas is a Senior Research Fellow and Deputy Director of the Bournemouth University Clinical Research Unit and an NIHR Research Design Service Consultant. Sarah has worked for many years in health services research, and her recent research activities include the development and manualisation of a non-pharmacological group-based intervention for people with multiple sclerosis who have fatigue, and this is called Facets, and also leading a pilot study called MyVitalise, which is exploring physiotherapist-supported home-based use of the Nintendo Wii for people with multiple sclerosis. So over to you, Sarah. Thank you.
Right, hi everybody. As Rona said, I'm going to be talking about the Facets, which is a fatigue management program that we've developed, and I'm going to be explaining how it's gone from research into practice. Now you might be wondering, why is it called Facets? And I thought it would be a good place to start. So it's called Facets, and that stands for fatigue, applying cognitive behavioural and energy effectiveness techniques to lifestyle. And hopefully the reasons why it's called that will become clear as my talk goes on. I'd like to first of all acknowledge the fabulous team who worked on this research. The chief investigator is somebody called Peter Thomas. I always have to point out he's not related to me. We share the same name. He happens to be my boss, but yeah, we're not related. I'd also like to acknowledge the two principal investigators, Paula Kirsten and Rosie Jones, and also say that Alison Knock and Vicky Slingsby were the occupational therapists who delivered Facets in the local centre, and they're now also involved in rolling out the programme. And also like to acknowledge Tim Warner and Jeff Linder, who are people with MS who've been involved with us throughout the research programme, and we're really fortunate to have Tim here speaking with us and sharing his experiences about fatigue in the Facets programme. So that's great. Just one more acknowledgement is to the other people who delivered Facets in the other centres, and of course to all the individuals who've taken part in this research. So what I'm covering today is the development of Facets, and that was funded by the MS Trust and the Bupa Foundation. I'm going to briefly describe the research trial that the MS Society has funded, and I'm going to be saying a bit about how the MS Society is supporting the rollout of the Facets programme. Now, I just wanted to say briefly, to echo Adrianne's talk, about the challenges that we face, not only in identifying, but also in measuring and managing fatigue. So fatigue is a really subjective phenomenon. I've got a roller coaster here to show how much it fluctuates and how variable it is for people. Even within the same day, it can vary. There's a Venn diagram there to show how it overlaps. So it overlaps with other symptoms. So, for example, depression, physical functioning, sleepiness. So it can be quite difficult to disentangle it from these other symptoms. And there's a complicated Rubik's type cube, to show that it has many facets. And as we've already noted, that it's an invisible symptom. So it's not like a broken leg where you have a plaster that's very obvious to people. So this all makes it very challenging when we're thinking about designing a fatigue management program. So how did we start going about developing Facets? Well, I think it's worth highlighting the time and effort that goes into developing an intervention. We started back, I think, in 2006, so it's definitely not an overnight thing. We were guided by the Medical Research Council framework for developing interventions. And developing the intervention involved the following parallel strands. So I firstly went and observed a local model of good practice, and that was a fatigue management program for people with a lifestyle management program for people with chronic fatigue syndrome. Now, clearly, fatigue is a major problem for people with chronic fatigue syndrome, and observing that program was really helpful, as there were some elements in that program that were relevant for our Facets program that we wanted to develop. At the start of the program, we also held a focus group with 8 people with MS, and that was mainly to run our ideas by them for a fatigue management program, to see what they thought of them. We wanted to ask about their experiences of fatigue, the impact it has on their lives, and what strategies that they find helpful. And this helped us to design the content of our program. We also got feedback about practical aspects too, such as where should we hold our program? What time of the day would be best? What do you feel about a group delivery? And should we involve supportive others? So the feedback from the focus group was quite reassuring for us and suggested that we were on the right track. Practical feedback from the focus group helped us to finalise practical details such as the venue, timing, involvement of supportive others. And participants expressed possible benefits of a group format. In fact, the strength of a group format seemed to emerge from the focus group itself. Participants who came to the focus group were swapping contact details at the end of it. And someone said, that's why I think a group like you're planning is good, because then a lot of MS people don't get to see any other MS people, and you sort of think, oh, it's just me. So if you get a group of people together and you're all having the same sort of symptoms, you can all talk about what's going on. So after that, we undertook a thorough literature search, not only of research in MS, but also in other chronic conditions, and also looked in self-help materials and books. And this is just a brief summary of the evidence base. So the evidence for drug treatments for fatigue is that they work for some people, but not for all people. And there's evidence that energy effectiveness approaches work fairly well, but that benefits often don't last. And cognitive behavioural therapy has been shown to be helpful in a wide range of conditions. So, for example, chronic fatigue syndrome and rheumatoid arthritis, where it's been used specifically for fatigue management. And psychologist-delivered cognitive behavioural therapy in multiple sclerosis has been found to be beneficial. However, this could be problematic to deliver, given that there are few psychologists in the NHS. So all of those strands led to the development of a draft manual, and all of that preparatory work helped us to decide on certain principles to guide the development of our new fatigue management programme. So we wanted to blend cognitive behavioural and energy effectiveness approaches because there was an evidence base for these being beneficial for people with MS fatigue, and I will say a little bit more about these approaches in a moment. We wanted to incorporate self-management. We didn't want to create a programme that people would come and attend for 6 weeks and that would be it. We wanted to provide people with tools and strategies that would help them, that would continue to help them in the longer term. We decided upon delivering the programme in a group approach, and this was for various reasons. The feedback from our initial focus groups, as I've said, indicated that quite a few people had never had a chance to speak with other people with MS. So it can provide a very normalising experience, realising that fatigue is a very real, common and debilitating symptom. Some people also commented that it's sometimes easier to accept advice from your peers than from a health professional, and so we wanted to encourage this process. And it's also potentially more cost-effective to deliver interventions in groups. We wanted the programme to be manualised so it would be easy to deliver, and we wanted it to be able to be delivered by occupational therapists, physios and nurses. Given the shortage of psychologists, we wanted to make sure that those people that typically deliver fatigue advice to people with MS could deliver it. And we also wanted to make sure that it could be rolled out easily in current health service structures. So when developing an intervention, it's important to make sure that it's based on theory, and we based Facets on the following approaches. So I've already mentioned self-management. Self-management recognises that living with a chronic condition is an ongoing process, and that the person with the condition possesses unique insights, expertise and knowledge. And that's never so true as in the instance of fatigue, where it's such an individual and variable symptom. And the person with MS fatigue is the best qualified person to try out a range of approaches and explore which methods of fatigue management best suit them. So we aim to tap into this expertise during the programme. Now, self-efficacy is similar to self-confidence. It's about people's confidence in their ability to manage their fatigue. And so we really wanted to encourage people's confidence. So we decided that we would include trying out fatigue management strategies as part of homework. And that way, people could experience successes in what they were doing and build their confidence. Also, another way self-efficacy can be increased is by encouragement from other members of the group. So we included flip chart activities and group work in our programme to encourage discussion among group members. Now, energy effectiveness is about using available energy in the most effective way. We know that people with MS have limited reserves of energy. And this is an approach often used by occupational therapists, and it involves planning daily activities, finding more efficient ways of doing them. One of the central features is achieving a balance between activity and rest. It can also involve modifying the environment. So I've got some examples here of energy effectiveness strategies. So, for example, I've tried to limit the things I must do in favour of what I want to do. So there's an example of prioritisation. Naps are a daily routine now and useful. So an example of resting. I think delegating has helped a lot. So an example, obviously, of delegation. I allow myself to rest as soon as needed, then complete my task later. So there's an example of pacing. Whereas before I was working like an idiot during my good time of the day, I now rest during the morning and I'm not so tired in the afternoon. So there we have an example of someone breaking that boom-bust cycle, which is quite common. However, while such approaches can be helpful, attitudinal barriers and emotions may stop individuals from applying them to their own lives. For example, it might be perfectly feasible for someone to change their daily routines and schedule rests into their day, but they will not give themselves permission to do so. And people with MS often experience strong emotions related to fatigue and fatigue management. Sorry, that's the barriers that people face. So here's some examples. I hate having to rest. I need to, but I hate doing it. I feel worse. If I rest, I feel worse. I feel guilty because I haven't done anything that day. So people feel a lot of guilt about fatigue, and the cognitive behavioral approach is helpful in getting people to become aware of and challenge pre-existing and sometimes quite long-standing expectations and thoughts related to fatigue and fatigue management. And we live in a society where much emphasis is placed on keeping busy and burnout is common. You can see this quotation here from someone about what can get in the way. Resting in the afternoon is very alien to me. That's what my granny used to do, but that's why I end up not being able to do what I want to do. Other cultures have different views towards rest. For example, in some places, a siesta in the afternoon is commonplace. So we can see in that example, unless thoughts and attitudes are considered, as well as the behaviours, change won't occur. And here's another example of how an attitude might get in the way of fatigue management. My own reluctance to give in, which often results in my overdoing it to the point of inducing severe fatigue, which exacerbates other symptoms. Sometimes people tell us that it's their own expectations that are actually their biggest barrier. People often have very high expectations of themselves and can be very harsh on themselves, much harsher than they would be, say, on a friend, and sometimes than they were on themselves even prior to their diagnosis of MS. And you can see how these kinds of pressures and expectations can lead to this boom and bust vicious circle. So people thinking, I must do as much as I can while I still can, and then overdoing it, paying for it later, leading to exhaustion, being wiped out, increased symptoms, and feelings of frustration and inadequacy. So what is the cognitive behavioral model? It can help to interrupt or break down these vicious cycles. It's concerned with individuals' attitudes and ways of thinking, that's the cognitive part, and what they do, that's the behavioral bit. And it's based on the theory that cognitions, emotions, and behaviors interact, and that sometimes changing how we think about a situation influences what we feel and what we do. So for those of you who prefer diagrams, this is a diagram of the cognitive behavioral model. And you can see thoughts, emotions, physical aspects and behaviors are all there continuously interacting. And when changes are made in any one of these elements of our lives, the other areas also change. So the cognitive behavioral approach is structured. It involves working in partnership, it uses problem solving, and it helps people to gain insights into how they think, feel, and behave. And it can help people to explore other ways of thinking and behaving that might be more helpful for managing fatigue. And to illustrate this point, this picture here shows how we can see the same thing or situations in more than one way. So you can see here, you can either see this picture as a bunny rabbit or as a duck, depending upon the way you look at it, even though the picture's the same. So okay, after all of that work, we developed our Facets program, and we then tried it out locally a couple more times. We ran 2 more groups. After the second group, we ran another focus group to get people's feedback, and then we were able to make final adjustments and refinements to our program. And so we ended up with our Facets resources. So we had a facilitator manual and a participant workbook, and we tried to use images and metaphors because they're quite easy to remember. And we had an accompanying PowerPoint presentation for each session, And we also built into the programme resources that were already available from the MS Society and the MS Trust, for example, Living with Fatigue and MS Essentials Fatigue, so that we were using resources that are available. So what are the objectives of Facets? The first objective is to increase people's understanding of fatigue. So that's really helped by meeting other people in the group. To normalise the experience, realise it's not laziness, that it's a very common experience in people with MS. To help people to use their available energy more effectively, so that's building upon those energy effectiveness strategies. And to help people to develop helpful thinking styles about fatigue. So that's using those cognitive behavioural approaches. So the structure of Facets is it's a 6-session programme held weekly. It's for groups of 8 to 10 people. The sessions build upon each other, and they're approximately 2 hours long with a refreshment break. People can bring a supportive other to the first session. That supportive other could be a partner, spouse, friend, relative, carer, employer, or a work colleague. We encourage informal contact outside of the group. Each session follows the same general format, so there's a facilitator delivered presentations, flip chart discussions, group activities and homework. At the end of each session, participants are given handouts to act as an aid memoir. Homework is included within the handout and it provides a resource for future reference. And in the facilitator manual, there are guidance notes for delivery for health professionals included. These are the sessions. As you can see, there are 6 sessions. The first one is what is MS-related fatigue. So that's a general introduction to fatigue, conveying that it's a complex thing. We ask about people's expectations of the programme. We have an icebreaker quiz. We look at contributing factors to fatigue, and we look at a conceptual model. The second session is about opening an energy account. For homework after the first session, people do an activity diary, so we get them to bring that back, and we look at whether they have examples of poor energy management. We also consider rest and the functions and barriers to resting, and we look at different types of relaxation, and we consider sleep as well. For homework after that session, people complete a rest, sleep, and activity planner and an energy measure. The next session is about budgeting energy and smartening up goals. So we're looking at different types of activity. What activities are draining and what activities are energizing for people? What types of exercise do people find beneficial? What are the barriers to doing exercise? How can people moderate their activity? And we give people guidance on setting realistic goals. The fourth session is about stress and the cognitive behavioural model. So in the fourth session, we introduce formally the cognitive behavioural model, and people have a thought diary for the homework. The fifth session is about challenging the unhelpful thoughts that they recorded in their thought diary. And the sixth session is about recapping and taking the programme forward. So we revisit the expectations that people described in the first session. We have a group activity. We play bingo to revisit the themes of the previous sessions, and we get people to plan their approach to fatigue management over the next 3 months. As you can see in the programme, there's a general shift from a practical to a more psychological orientation. But even in the first session, we introduced the cognitive behavioural elements. So that is getting people to think about their fatigue in terms of thoughts, emotions, behaviours, and physical aspects. And that helps the participants to get used to and start to feel comfortable with identifying and thinking about the links between the cognitive behavioural model. And that seems to work well, introducing it in that way, because sometimes people can feel quite overwhelmed if you introduce a psychological model straight away. So there we were. We'd developed our programme. We tried it out locally a couple of times. We were then just about to run our large trial, but first we ran a small pilot study. That was a dry run-through of our large trial to check our processes were going to work. So the Facets trial took place in 3 UK centres. People were randomised to either the Facets programme plus their usual care or usual care. The inclusion criteria were a diagnosis of MS, significant fatigue, and ambulatory. The research questions were, does Facets improve fatigue levels, confidence in managing fatigue, and quality of life? So we measured fatigue, quality of life, self-efficacy, and mood to try and capture that multifaceted nature of fatigue. And we got people to complete questionnaires that were sent to them in the post before Facets, 1 month after Facets, and 4 months and 1 year later. So what did we find? Well, we found that fatigue, quality of life, and self-efficacy were changed, but we didn't find any changes with mood. So Facets is an effective method to help people affected by MS manage their fatigue and increase their confidence to manage it. And at 1 year follow-up, we also saw improvements in quality of life. Overall, participants who attended the Facets programme seemed to be very satisfied with it. And this finding that quality of life change didn't happen until 1 year later seems to make sense because lifestyle changes take a while to implement. So I've just got a couple of quotations here from people who attended the programme. Before I used to battle with it, fatigue, convinced that I could beat it. But since taking this Facets course, I've realised that perhaps I can't beat it, I can manage it. And so this shows self-efficacy and confidence in managing fatigue. Sometimes people can exert a lot of energy trying to fight against it, battle with it, and that's not always, that doesn't always give you control, really. So I think that's an interesting quotation. Another one here. The Facets course made me realise I didn't have to feel guilty for not being able to do everything I used to do. I've learnt to say no. I've slowed down and am able to lie down in the afternoon and relax. And I think in just those couple of sentences, there are an awful lot of changes that person has made. So they've now learning to say no. They've changed their expectations. There's some acceptance. They're being less critical of themselves, giving themselves permission to do things differently, using pacing, incorporating relaxation techniques, and scheduling rests. So, okay, after we ran our large trial, there seems to be from Facets some benefits for people with MS in terms of fatigue management. But as researchers, we don't really have the time, resources, or expertise to do anything about rolling these kinds of research findings out and getting the benefits out to people with MS. So what often happens with research is it ends up languishing, getting dusty on a shelf, and you definitely don't want that to happen. And we've been very fortunate in that the MS Society has supported us to roll out the Facets programme. And thanks to people from the MS Society like Jude and Meg and Rona, they've been brilliant in helping that process happen. So we had quite a sorry state of a manual that we developed with photocopies, and you don't want to spend too much money on developing something until it's shown to be beneficial. And it was like a changing rooms scenario on the television where you have a room in your house and they come in and they make it all look nice. Well, the MS Society did that with our manual, so we can see that they've produced a nice fancy manual there for us. And the MS Society has been supporting and organising one-day Facets training courses for health professionals. And these have been delivered by Alison Knock and Vicky Slingsby. They're doing a fantastic job. And there's Alison Knock there in action. And they've been training in London, Glasgow, Manchester and Belfast. And if anyone's got any questions about that, Rona would be the person to ask. And to date, I think 99 health professionals have been trained. If you're interested in accessing Facets, if this appeals to you, then and you're interested in attending a programme, speak to your local healthcare professional to see if it's currently available in your area. If it isn't available, ask your healthcare professional to contact the MS Society for further information. And just an update that we're quite excited because Facets has now been translated into French, and we've got some colleagues in France who are hoping to develop some booster sessions to try to encourage the maintenance of behaviour change from people who are attending the programme. It's in the process of being translated into Norwegian and German, and we've just submitted our one-year follow-up results to an academic journal, so fingers crossed with that one. So there's some references there if anyone's interested, and the main paper reporting on this research is open access, so if anyone's interested you can easily access it, and I think there's a link to the paper possibly on the MS Society website. And just like to thank everyone for your time. Thank you.
Thank you, Sarah. The third speaker this afternoon is Tim Warner, and Tim's going to talk about his experience of Facets and the strategies that he uses to enable an active lifestyle. Tim has MS and was diagnosed a number of years ago. He gave up work about 12 years ago, and since then has had what I would say is an incredibly active lifestyle with many, many activities which you'll hear about. And I'll hand over to Tim to talk further.
Thank you, Rena. Right, thank you. Good afternoon, everybody. I do lead quite an active lifestyle, probably more so since I've given up work, doing things I enjoy, getting a good feel-good factor from them. This includes things like horse riding, shooting, dog walking, sailing, all sorts of things. Not on one day at all, but all sorts of things. And flying, everything you can think of, I just do it. So my fatigue, I realise it's very different for everybody, but for me it can be very overwhelming, meaning I can't do anything else, I can't think of anything. It just stops me dead in my tracks, and I just have to stop. I get grumpy because I can't do things, and angry and frustrated because I can't do things. But I am getting better at planning things and making strategies, and also learning when possibly my fatigue is going to hit me so I can plan around it. How I was involved with Facets was I was invited by Sarah and Peter Thomas and the OTs at Poole MS Service to join the pilot study, which I found very interesting. I learned a lot myself from the actual course itself, and the pilot study itself, and from talking to other people. There were 8 others on my group, 2 people I knew very well, the other people I didn't know at all. Learning their understanding of fatigue, them understanding my understanding of fatigue, and sharing ideas. Then I was invited by Sarah and Peter Thomas to join the steering group, which I was on for a number of years, with Jeff Linder, another person with MS, and again that was very interesting. I'd like to think that I helped with my input, with some ideas and some corrections of things and changes and things such as that. And I learned a lot more from the pilot studies and steering groups, meeting other people, how understanding my fatigue more. My strategies. I do write a lot of lists and plan and things out, and as I get things done, I'll cross them out so I know I've done them. If I don't get them done, they'll go on to the next day, and I try and prioritise what is important that day to get done, and try and get them done, and carry things over. Sometimes I don't get it done, and I determine to get it done maybe, and I end up, Sarah calls it boom and bust, I call it crash and burn, where I just totally can't take any more. I ensure I have a good night's sleep, fairly religiously. It's sort of bedtime between 10 and 10:30, have a good night's sleep, get up sort of 7:30, 8 o'clock in the morning, and make sure I sleep well. I avoid sleeping during the day. I find if I do fall asleep during the day, I wake up from a heavy sleep and I feel very much worse for it, so I avoid actually sleeping during the day. But I do take rest breaks where I sit in the front room or conservatory or something like that and just listen to the radio a bit and just have some quiet me time. I try to avoid secondary fatigue, what I call secondary fatigue, which is losing the feel-good factor. I look very carefully at medication, what the side effects of it, if anything's going to cause tiredness on that. I try to eat a good balanced diet, and any other cause that make me feel tired, and too much stress I try to avoid as well. And that's it really for me. Thank you much.
Thank you. I'd like to thank all our speakers, and I'd like to invite anyone in the audience to, if they have a question, could they possibly put their hand up? We do have some roving mics, and if you can put your hand up, and then we'll get a microphone over to you for a question. There's a gentleman down there. Okay.
I asked a question before. How do I find out where Facets is being rolled out? I did ask my MS nurse, and she'd never heard of it.
Okay, well, I'll answer that one. If we are trying to run a number of Facets courses over the year, and at the moment we have certainly 2 planned for this year, another 2 in the pipeline. If your MS nurse or any MS professional contacts us at the MS Society, we will let them know where they are, and we will do our best to help them attend one that's going to be local to them.
And how did it take place? I did ask the question to Adrienne before. Are they residential?
It's a 1-day course, a training day that they come to. It's a full day. We make them work very hard all day, but it's just 1 day that they will come to. And at the end of the day, they will have gone through what they will need to do for running out the programme.
Right, thanks.
Are there any other questions? I'm sure somebody must have you. Lady down the front, very front. Here, please, in the middle. Sorry, the microphone's just coming to you. Sorry.
Hello, how is this Facet programme funded? Does the individual pay for it, or does it come out of the MS professional nurses budget?
For them coming on to do the training day, we have to charge for them to come to do the training day, but we will offer them, they can apply for a study grant through the education department, which will cover up to 50% of the fee for the day. And usually the other 50% is covered by their employers. Sometimes some of the local branches of the MS Society have actually been able to support the people, the professional who wants to do the course, with the remainder of the funding.
What about the individual who wants to attend the...
Do you mean any
I'll hand that over to Adrienne.
MS patients. What we've done in our area is we've got a long-term neuro team and the MS nurse myself, and most places, most health authorities or trusts do put on some sort of symptom management or fatigue programme anyway, and it's a case of it's usually built into what we actually do for MS. So what we do is we roll it out as we do them twice a year. Our first Facets was in October last year, so we're looking to do another one, and it's a case of venues and what time of day we're going to do it to suit the MS patients, basically. But it's usually incorporated, and obviously it's free because it's a point of contact for us. But again, some of the MS Society branches, mine in particular, will fund us to rooms to provide this sort of programme.
It's quite interesting to add that I know that they're delivering it in a hospital in London, and they're finding that the people there find it easier to attend an evening session, and they're actually running evening sessions. So it varies as to what happens. That's Susan Hurahan?
Yes, Queen Square.
Queen Square.
Gentleman at the back.
I speak as a branch manager. We're happy to fund these courses in our area. We tried the evening, but we found that people were too fatigued, so we've gone back to the daytime. Just one thing is, it does seem to be making a difference to some people, but I guess the jury's still out on a long-term basis. I mean, a year down the road, we haven't seen any evidence whether this is being highly successful or partially successful or whatever. Are you still amending the course as you find out more about how it's being rolled out? Or is it fixed for all time now?
I'll let Sarah answer that one.
That's a tricky question. We have run a kind of definitive trial, really. We did a lot of research trials don't have very long-term follow-up, but we did follow up for a year and found that benefits were largely being maintained at 1 year. But I'd be interested to chat with you about your thoughts on, where you think improvements could be made. Be interesting. It's always difficult as to where you put the finishing point, really, with things, because you could amend them forever. Yeah. And it's the issue about the evidence base. But yeah.
I also think I used to run my own fatigue programmes in the past that were sort of my made-up ones, obviously research-based. And what I found is that some of my patients have now come on the Facets programme, so it'd probably be quite interesting for me to actually look who benefits more from sort of like a structured programme like Facets or the sort of made-up ones. And I know made-up sounds really unprofessional, but I think you know what I mean. And it might be interesting for us to sort of work on how we see, but again, I'm only like two sessions down the road as well.
Yeah, and I think, it's variable, it's not one thing, one size fits all, really. It's some people, group work suits better, other people, like you say, where you work, you do lots of home visits, so.
Maybe as a follow-on to that one, I don't know if Adrian or Sarah would like to answer, maybe even Tim. It's once you've done the course and you're putting all the various strategies into practice, how do you actually manage to, Tim, how do you manage to keep doing them? Adrienne and Sarah, how do you manage to keep encouraging people to do them?
You first.
thank you. I think it's very much sort of by doing a course over so many weeks, it's very much instilled into your mind and you learn the strategies and you keep continuing to use them, modify them slightly to your own needs, but you use the strategies that you share with other people and learn from the course and continue to use them as you see fit and as you require them.
And from my point of view as a professional, when I see somebody sort of dropping or getting problems again, I remind them about the workbooks, because you get all the workbooks from session 1 to session 6, and each workbook's got the subject that you've been doing, and in session 6 you get a summary of everything that you've learnt, so the tools that you've acquired over the 6-week programme, and that I sort of very sort of tongue-in-cheek said, stick that to a kitchen cupboard, because then if you're failing on SMART goals or pacing or relaxation, it actually tells you which workbook you've looked at. So I tell them to go away and read it. So I'm constantly saying, read number 2, read number 5, read number 6. And I'm finding that patients are adapting to me badgering them a little bit.
And I think we have got some qualitative work that we did that we haven't published yet, but we did get quite a few people saying that those resources are quite good to revisit in the future. And, we all have good intentions, but maybe circumstances change. So 8 months later, when maybe you've had a person's had a relapse and things have changed, to revisit them, and it's quite helpful for people. Also, as I think I said, our colleagues in France are looking at developing some booster sessions, and I think, again, that can help people to keep the momentum going. If you don't have that built-in support like it sounds like you have.
Yeah, it's booster sessions I was thinking about.
Yeah.
I'd like to know how you, once they've gone from the 6 weeks, do you find out, how do you find out a year down the road, is it still making a difference to their lives? You get the group back, Or do the MS nurses just meet them
I
individually?
think it would be up to the MS nurses in your area or whoever was running it, whether they wanted to. I'm planning a year follow-up on all the ones that I've done it with, but that is because I've got that capability, because my practice is purely 100% home visits.
Yeah.
So I mean, some of my colleagues will think that's lucky or not lucky because I don't see people in clinics because I'm purely community. So I have got that opportunity, but I would imagine it would depend on the health professional, MS Society branch, to probably do that as their own project.
Right, thank you. We can talk more about this.
Yes, I'd love to, and maybe there's a research project in here somewhere.
Are there any other questions? No last burning issues about fatigue and its management? No? Well, I'd like to take this opportunity to thank you all for coming along to the session. I'd like to especially thank our 3 speakers today and all the information they've given. Please do remember, if you require any information about fatigue and its management, we do have information at the hub, and you can also download any of the information either from the research pages on our website or the publications themselves. If you have an MS nurse or if you see a physiotherapist or an occupational therapist, do ask them about fatigue and its management, and if they offer any courses, whether it's on a one-to-one basis or a group. And if they aren't offering anything, do refer them back down to us. And enjoy the last little while that you have of MS Life 2014. I think there's about an hour to go, so go out and enjoy the last hour, and thank you very much for attending. Thank you.